Adenocarcinoma of the stomach and other gastric tumors. Michael Quant, Jan Bornschein 54. Gastric cancer remains a major cause of cancer-related mortality in the world, despite declining rates of incidence in many industrialized countries. In this chapter, we mainly discuss gastric adenocarcinoma, which makes up most of gastric malignancies. Epidemiology. Gastric cancer is the third leading cause of cancer mortality in the world. Although the overall incidence is declining. 2 In Western countries, the incidence of gastric cancer has decreased dramatically over the past century. In the USA, gastric cancer mortality has decreased 87% since 1950, with a similar trend being reported in Europe. 3 In the USA, the incidence of gastric cancer has diminished to approximately 7.6 cases per 100,000 people, whereas as recently as 1945, gastric cancer was the leading cause of cancer mortality in men. 5 There is great geographic variation in gastric cancer incidence, with the highest incidence rates in the Far East, Fig 54.1. Eastern Europe and Central and South America also have high incidence rates, with the lowest incidence rates observed in North America, North Africa, South Asia, and Australia. 6 Although gastric cancer was common in industrialized countries in the past, the latest epidemiologic data indicate that more than 70% of new cases of gastric cancer are in developing countries, reflecting a more rapid decline in developed countries. 1 In the USA, the median age of diagnosis is 70 years. 4 In Japan, a country with a high incidence of gastric cancer, the mean age of diagnosis is roughly a decade earlier, perhaps reflecting lead time bias due to widespread screening. The incidence of gastric cancer in males is approximately twice that in females. Table 54.1.1 The incidence of gastric cancer in blacks in the USA is nearly double that in whites. Native Americans and Hispanics also have a higher risk of developing gastric cancer than whites. In contrast to the pattern seen with non-junctional gastric cancers, the incidence rates of adenocarcinomas at the esophagogastric junction, EGJ, formerly cardiac cancer, are rising too, according to the U.S. Surveillance, Epidemiology, and ENDS results, SEER, database. These junctional cancers now represent 27% of gastric cancers in the USA, up from just 10% in 1975. 4 There are numerous dietary, environmental, and genetic risk factors for gastric adenocarcinoma. Box 54.1. The dominant risk factor remains, however, infection with HP and the associated chronic active inflammation of the gastric mucosa. See Chapter 52. Etiology and pathogenesis. Gastric cancer can be subdivided using the Laurin classification into 2 distinct histologic subtypes with different epidemiologic and prognostic features. Fig 54.2.7 The intestinal type of cancer is characterized by the formation of gland-like tubular structures with features reminiscent of intestinal glands. This type of gastric cancer is more closely linked to environmental and dietary risk factors, tends to be the predominant form in regions with a high incidence of gastric cancer, and is the form of cancer that is now declining worldwide. The diffuse type of cancer lacks glandular structure and consists of poorly cohesive cells that infiltrate the wall of the stomach. It is found at the same frequency throughout the world, occurs at a younger age, and is associated with a chapter outline epidemiology 820 etiology and pathogenesis 820 HP infection 823 dietary risk factors 825 cigarette smoking 825 alcohol 825 obesity 825 genetic factors 826 tumor genetics 827 premalignant conditions 829 chronic atrophic gastritis 829 intestinal metaplasia and dysplasia 830 gastric polyps 831 previous gastrectomy 831 PUD 831 Menetrier disease 832 screening and surveillance 832 prevention 832 eradication of HP 832 aspirin and other NSAIDs, including COX-2 inhibitors 833 statins 833 antioxidants 833 other dietary factors 834 clinical features 834 Diagnosis 834 Endoscopy 834 CT Gastrography 834 Serum Markers 835 Classification and Staging 835 EUS 836 CT and PET 836 Laparoscopy with peritoneal lavage 837 Other imaging modalities 837 Rest aging after neoadjuvant treatment 837 Prognosis and treatment 837 Surgery 837 Endoscopic mucosal resection and submucosal dissection 838 Chemotherapy 838 Chemoradiation 839 Intraperitoneal chemotherapy 840 Unresectable disease 840 Miscellaneous gastric tumors 841 820 Chapter 54 Adenocarcinoma of the stomach and other gastric tumors 821 54 Worse prognosis than the intestinal form. Extensive involvement of the stomach by the diffuse type can result in a rigid and thickened stomach, a condition referred to as linitis plastic, video 54.1. Another key feature of diffuse type cancers are signet ring cells, special mucin-filled cells that are not present in intestinal type adenocarcinomas. There are also mixed phenotypes that contain heterogeneous areas that feature predominantly either intestinal or diffuse type characteristics. Adenocarcinoma of the stomach is also classified into proximal tumors, EGJ and gastric cardia, and distal or non-junctional tumors, fundus, body, and antrum of the stomach. Junctional cancers can be further classified according to the Seward classification by the location of the main tumor mass into type I, 1 to 5 cm above the EGJ, type 2, from 1 cm above to 2 cm below the junction, and type 3, 2 to 5 cm below the junction, tumors. 8 There is no clear distinction between the genetic and cellular origin of adenocarcinomas of the distal esophagus, the EGJ, and a subgroup of non-junctional distal gastric cancers. 9 Interestingly, with the decreasing incidence of HP infection, non-junctional tumors have been declining while more proximal tumors have been increasing. In a mouse model, it has even been postulated that Barrett esophagus-related esophageal cancer and cancer of the EGJ have their origins in the gastric cardia. 10 Emerging data from gene expression profiling suggest that differences in pathologic appearance and clinical behavior may be due to the presence of unique molecular phenotypes. Characterization of the gastric cancer genomic landscape reveals the presence of multiple alterations in the expression of tyrosine kinase receptors, which in conjunction with their ligands and downstream effector molecules represent potential pathways for future drug development. The Cancer Genome Atlas, TCGA, consortium suggested 4 gastric cancer subtypes based on the genomic profile of about 300 gastric cancers. 11 This classification correlated well with the clustering of high-throughput data of different platforms including epigenome, transcriptome, and proteome analysis. So far, there are only little data to support the biological relevance of this proposed classification. Previous transcriptome analyses of gastric cancers, on the other hand, have demonstrated phenotypic clusters with either distinct prognostic outcomes or different response to systemic treatment. 12-13 It is believed that the development of intestinal-type gastric cancer occurs through a multi-step process in which the normal mucosa is sequentially transformed into a hyperproliferative epithelium, followed by metaplastic processes leading to glandular atrophy, dysplasia, and then carcinoma. In colon cancer, the evidence is strong that each step in the transition is associated with a specific gene mutation. 14 But the evidence that gastric cancer follows a comparable sequence of genetic events has been lacking. However, in both the intestinal-type gastric cancer and colorectal cancer, it does appear that DNA mutations are established over time in stem cells in the normal human stomach, and that in intestinal metaplasia these mutations spread through the stomach through a process involving crypt fission and monoclonal conversion of glands. 15 The contention that the pathogenesis of intestinal-type gastric cancer is a multi-step process is supported mainly by the observation that both chronic atrophic gastritis and intestinal metaplasia are found in higher incidences in patients with intestinal-type cancer and in countries with a high incidence of gastric cancer. See Chapter 52. 16-17 This multi-step model of intestinal-type gastric cancer, developed in large part by Palaio Correa and colleagues. 18 postulates that there is a temporal sequence of paraneoplastic changes that eventually lead to the development of gastric cancer. A common feature of the initiation and progression to intestinal-type gastric cancer is chronic inflammation of the gastric mucosa. HP infection is the Table 54.1 gastric cancer incidence and mortality rates per 100,000 population, age-adjusted. In 2012 incidence mortality male-female developed countries 15.66.79.24.2 developing countries 18.17.8 14.46.5 data from Torre LA, Bray F, Siegel RL, ETAL Global Cancer Statistics, 2012. CA Cancer J. Clin Internet. 2015. 65:87-108. Available from http://www.inspi.nlm.nih.gov/pubmed/2565178 Fig 54.1 Worldwide incidence per 100,000 population of gastric cancer in men in 2012. WHO 822 Part 6 Stomach and duodenum primary cause of gastric inflammation and the leading etiologic agent for gastric cancer, see Chapter 52. In a subset of patients, the inflammatory process leads to the development of atrophic gastritis, with loss of glandular tissue, followed by progression to intestinal metaplasia, dysplasia, early gastric cancer, and, eventually, advanced gastric cancer. Fig 54.3. Although animal models suggested that all stages prior to the development of high-grade dysplasia are potentially reversible, there is still ongoing debate what defines the point of no return for humans from which further progression of neoplasia can no longer be prevented. 19,20 Eradication of HP has the potential to prevent gastric cancer as shown in recent meta-analyses. 21,22 The preventive effect of eradication is more evident if there are no paraneoplastic conditions of the gastric mucosa, glandular atrophy, intestinal metaplasia. At the time of intervention. 23 HP eradication can prevent further progression of paraneoplastic conditions, and even a certain degree of regression can be documented. 24 Although it is currently assumed that presence of intestinal metaplasia is most likely to mark the point of no return, there is even an effect of HP eradication if advanced lesions are present, e.g., after endoscopic resection of an early gastric cancer. 25 Unlike the situation observed with colon cancer, the precise genes involved in each step of this progression are still not defined. Nevertheless, next-generation sequencing techniques have shown that there is more heterogeneity in genetic alterations in gastric cancer and cancer of the EGJ than in colon cancer. 26,27 Furthermore, the premalignant stages of gastric cancer are not as readily identifiable during endoscopy as those of colon cancer, and many gastric carcinomas are very heterogeneous, containing a large percentage of stromal cells. These stromal cells, which also include cancer-associated fibroblasts known to promote tumor growth, have been reported to show distinct genetic and epigenetic changes that may confound tumor analysis. 28,29 This feature makes characterization of the timing of specific gene mutations in gastric cancer difficult at best. Currently, the role of chronic inflammation in the diffuse type of gastric cancer, as well as the similarities if any to the proposed pathway in Fig 54.3 for the intestinal type of cancer, remain to be clarified. One common factor that is related to both histological subtypes is a strong association with HP infection, which has shown to directly modify genes involved in DNA damage repair, DDR, pathways. 30 Modifications of DDR-related genes are a common event in gastric carcinogenesis. 31 Box 541 Risk factors for gastric adenocarcinoma Definite adenomatous gastric polyps * Chronic atrophic gastritis Cigarette smoking dysplasia * EBV History of gastric surgery, especially bilirubin 2, * HP infection. Intestinal metaplasia Genetic factors Family history of gastric cancer, first-degree relative, * familial adenomatous polyposis, with fundic gland polyps, * Hereditary non-polyposis colorectal cancer juvenile polyposis Peutz-Jeghers syndrome probable high salt intake history of gastric ulcer obesity, adenocarcinoma of the cardia only, pernicious anemia regular aspirin or other NSAID use, protective, snuff tobacco use possible diet high in nitrates heavy alcohol use high ascorbate intake, protective, high intake of fresh fruits and vegetables, protective, low socioeconomic status Ménétrier disease statin use, protective, questionable high green tea consumption, protective, hyperplastic and fundic gland polyps surveillance for cancer is recommended in patients with this risk factor. Fig 54.2 Histopathology of the two types of gastric cancer. A. The intestinal type of gastric adenocarcinoma is characterized by the formation of gland-like tubular structures mimicking intestinal glands. B. The diffuse type of gastric cancer contains singly invasive tumor cells that frequently contain abundant mucin and that lack any glandular structure. H&E Stains. Courtesy Rhonda K. Yantis, M.D., Boston, Massachusetts. AB Chapter 54 Adenocarcinoma of the stomach and other gastric tumors 823-54 HP infection. See also Chapter 52. HP is a gram-negative microaerophilic bacterium that infects nearly half the world's population and is recognized as the primary etiologic agent for gastric cancer. Indeed, H. pylori has been classified as a Class I, or definite, carcinogen by the International Agency for Research on Cancer, a branch of the WHO. Infection with HP has been found in every population studied, although the prevalence is higher in developing countries and most parts of East Asia. 32-33 The natural history of chronic HP infection includes three possible outcomes. 34, 1. Simple gastritis, where patients often remain asymptomatic. 2. Duodenal ulcer phenotype, which occurs in 10% to 15% of infected subjects, and 3. Gastric ulcer/gastric cancer phenotype. The risk for gastric cancer development varies with the type of background gastritis, but in general, corpus-dominant gastritis resulting in a low acid state is mainly associated with an increased risk. HP-induced duodenal ulcer disease is associated with a high gastric acid output as well as a reduced risk for developing gastric cancer. 35 Studies suggest that HP-infected patients develop chronic atrophic gastritis at a rate of 1% to 3% per year of infection. 1836-37 Thus, those patients who are genetically predisposed to developing atrophic gastritis in response to HP infection are likely to be also predisposed to gastric cancer. Although HP infection is associated with both diffuse type and intestinal type adenocarcinomas, we focus in this chapter mainly on the mechanisms responsible for the formation of intestinal type adenocarcinoma because they have been better studied. The association of HP with mucosa-associated lymphoid tissue lymphoma is discussed in Chapter 32. The increased risk of development of gastric adenocarcinoma due to HP infection depends on multiple factors including host genetic factors, the strain of bacteria, including bacterial virulence factors, the duration of infection, and the presence or absence of other environmental risk factors, e.g., poor diet, smoking. In a Japanese cohort, only those infected with HP developed gastric adenocarcinoma during follow-up, 2.9% vs. 0%, p. 0.001. 38 Additional cohort studies from China and Taiwan have reported similar findings. 39,40 In Western countries, the association between HP and gastric cancer appears to be confined to non-junctional tumors. 41 There are data, however, that HP is likely to be also associated with Seward type 3 junctional cancers as well as potentially a subgroup of type 2 tumors. 42,43 A combination of a virulent bacterial strain, a genetically permissive host, and a favorable gastric environment may be necessary for cancer to occur. Currently, genetic susceptibility factors of the human host are studied based on individual genes, but new technologies such as next-generation sequencing will enhance the identification of host genetic factors. Nevertheless, the most important factor appears to be the induction of chronic inflammation by HP infection. This leads to an impairment of the epithelial barrier function of the gastric mucosa, thereby increasing the impact of other pathogenic factors, e.g., diet. Several aspects of the inflammatory milieu have been implicated as carcinogens. They include increased oxidative stress and the formation of oxygen-free radicals, leading to DNA damage, increased CD4+ T cells and myeloid cells, and elevated pro-inflammatory cytokine production, all leading to accelerated cell turnover, reduced apoptosis, and the potential for faulty or incomplete DNA repair. 44 Indeed, recent studies suggest that animals with deficient DNA repair mechanisms display more severe gastric dysplasia after chronic infection with HP. 45 As mentioned earlier, HP is capable of directly modifying DDR-related genes and their function. 30 Thus, evidence to date clearly indicates that the most important cofactor in the induction of Helicobacter-related disease is the host immune response. Indeed, chronic inflammation has been linked to a large number of non-gastric cancers. Chronic inflammation of the gastric mucosa appears necessary for the progression through atrophy to gastric cancer. Disease mechanisms are difficult to study in human infection, and therefore, much of our understanding of the immune response to Helicobacter organisms comes from work performed in a mouse model. Different inbred strains of mice respond to infection with varying degrees of disease susceptibility, and several knockout models have helped to elucidate the roles of individual components of the immune response in disease. Genetic manipulation of the C57BL/6 susceptible murine strain has facilitated detailed study and has thus led to a deeper understanding of genetic factors that promote murine gastric cancer, and particularly, the role of the adaptive immune response. For example, gastric Helicobacter infection in mice deficient in lymphocytes does not result in tissue damage, cell lineage alterations, or the metaplasia-dysplasia carcinoma sequence. 46,47 In contrast, infection in B-cell deficient mice, which retain a normal T-cell response, results in severe atrophy and metaplasia identical to that seen in infected wild-type mice. 47 Taken together, these studies underscore the crucial role of CD4+ T lymphocytes in orchestrating gastric neoplasia. Susceptible mouse strains, such as C57BL/6, mount a strong Th1, T helper cell type 1, expressing interferon IFN-gamma and interleukin IL-12, type of immune response, whereas resistant normal gastric mucosa chronic inflammation dysplasia metaplasia atrophic gastritis superficial gastritis carcinoma H. pylori diet, salt. Recruitment of BM cells that form a tumor microenvironment salt. N-nitrosocarcinogens chronic inflammation and ROS higher gastric pH bacterial overgrowth and nitrate reduction barrier defect Fig 54.3 proposed chorea pathway of pathologic events in gastric adenocarcinoma. In well-differentiated, intestinal-type gastric cancer, histopathologic studies indicated that chronic HP infection progresses over decades through stages of chronic gastritis, atrophic gastritis, intestinal metaplasia, dysplasia, and cancer. The development of cancer has been attributed to alterations in DNA caused by chronic inflammation, which is associated with the recruitment of bone marrow-derived immune and mesenchymal cells, BM cells, that form a microenvironment that favors tumorigenesis. An imbalance between epithelial cell proliferation and apoptosis and, in a milieu of atrophy and achlorhydria, gastric colonization by enteric bacteria with nitrate reductase activity facilitating formation of carcinogenic nitrosamines allow the accumulation of oncogenic genetic alterations. Corpus predominant atrophy, or the loss of specialized glandular cell types such as parietal and chief cells, appears to be the critical initiating step in the progression toward cancer. From Fox J.G., Wang T.C. Inflammation, Atrophy, and Gastric Cancer. J. Clin Invest 2007, 117:60-9. ROS, Reactive Oxygen Species. 824 Part 6 Stomach and duodenum strains, such as the BALB/C, have a polarized Th2 response, expressing IL-4 and IL-5. 4858 Th2 response is associated with protection from mucosal damage despite the inability to eliminate bacterial colonization and, in fact, is often associated with higher bacterial colonization rates. Mouse strains such as the C3H, which has a mixed Th1/Th2 cytokine profile, show intermediate disease, suggesting that cytokines within an immune response interact to form a continuum of disease rather than discrete disease states. More recently, Th17 cells, expressing IL-17, have been shown to be an important component of HP-induced gastritis. 51 Although the composite immune milieu most likely dictates disease manifestations, there may be a role for individual cytokines in both the predisposition to and protection from disease. During Helicobacter infection, the Th1 cytokine IFN-gamma can promote or inhibit inflammation-driven cancer of the stomach, suggesting that a more specific immune response is responsible for cancer promotion or surveillance. Although studies in the past have suggested that IFN-gamma might promote the development of gastric preneoplasia, 52 IFN-gamma overexpression in the stomach at low levels was recently shown to be able to suppress gastric cancer in models of IL-1 beta and Helicobacter phellis-dependent carcinogenesis. 53 In addition, IFN-gamma was shown to counteract the development of Th17 cells. 53 Thus, different composition of the same cells and cytokines in the tumor microenvironment can contribute to a constellation that favors or inhibits carcinogenesis. On the other hand, mice lacking IL-10, a cytokine that acts to dampen an immune response, demonstrate severe atrophic gastritis in response to infection. 4650 More recently, genetic murine models have illustrated the importance of the IL-6/IL-11 family of cytokines in the development of gastric cancer. 54 Manipulation of the immune response within wild-type strains confirms the central role of the Th1/Th2 response in producing disease. For example, infection with the intestinal helminth Heligmosomwates polygyrus skews the immune response toward Th2 polarization and protects the C57BL/6 host from Helicobacter-induced atrophy and metaplasia. 55 This mouse model mimics both the parasitic infection status and the paradoxical low gastric cancer high HP infection rates seen in areas of Africa, potentially explaining this apparent inconsistency. These observations in mice led to human studies in Africa and Latin America that confirmed that geographic regions with low gastric cancer rates had much higher Th2/Th1 immune responses to HP. 56,57 In general, the increased Th2 type responses were found in areas where serum IgE levels were high and the prevalence of intestinal parasitism by helminths is above 50%. These findings further stress the importance of the host response to infection and suggest the possibility that manipulation of the genetically predetermined host cytokine profile in response to environmental challenges may lessen or exacerbate the disease process. Studies on human tissue have also demonstrated that the degree of colonization with HP depends on various factors, such as the presence and activity of regulatory T cells, TREG, or the initial, naive, parietal cell mass, which reflects the acid-secreting capacity of the gastric body. 5860 TREGs are associated with increasing bacterial colonization 61 chronic inflammatory changes 62 and the expression of immunosuppressive cytokines like IL-10, IL-17 and TGF-beta. 59,63 In case of gastric cancer, TREGs are increased both in the gastric mucosa and the peripheral blood. 6466 The ratio of Th1/Th2 derived cytokines is the highest in asymptomatic gastritis, showing a steady decrease in gastric atrophy, intestinal metaplasia, and intraepithelial neoplasia progression to gastric adenocarcinoma. This is associated with a concomitant increase of the TREG cell compartment in the peripheral blood as well as persistence of CAG-A-positive strains that favors a TREG-mediated chronic inflammation. 67 Whereas HP infection has been unequivocally linked to gastric cancer, the development of dysplasia and invasive cancer tends to occur at a time when HP colonization has either dramatically declined or, in some cases, has disappeared from the stomach altogether. Gastric cancer almost always occurs in the setting of prolonged gastric atrophy and hypochlorhydria, a condition that predisposes to enteric bacterial overgrowth. Although antibiotic eradication therapy targeting HP delays and inhibits development of gastric cancer in mice, 20,68 antibiotics eradicate not only HP but also other microorganisms that colonize the atrophic, Hypochlorhydric stomach. Indeed, infection of otherwise germ-free INS gas mice with HP resulted in delayed progression to gastric cancer compared to HP-infected INS gas mice colonized with conventional flora. 69. Thus, HP may represent simply the initial, or the most prevalent, microbial factor responsible for gastric cancer progression. Ferreira et al. Reported recently that the gastric microbiota of patients with gastric adenocarcinomas is significantly different from patients with chronic gastritis. 70. The dominant gastric dysbiosis is characterized by reduced microbial diversity and reduced HP abundance, as well as an overrepresentation of bacterial genera that include intestinal commensals. With regard to these findings, it is also of interest that HP when present dominates the otherwise much more diverse gastric microbiome in humans, as well as mice. Prior to cancer development. 71. There is a great deal of genetic diversity between strains of HP owing to point mutations, insertions, deletions, and base pair substitutions within the genome. Several strains may infect a single individual, and existing strains can undergo mutations and change over time. 72,73. Despite this genetic diversity, several genes are recognized as risk factors for gastric carcinoma, including the CAG pathogenicity island, the VACA gene, and the BABA2 gene, being the most relevant and most extensively studied thus far among other factors. The HP genome is 1.65 million base pairs and codes for approximately 1,500 genes, two-thirds of which have been assigned biological roles. 74. The function of the remaining one-third of the genome remains obscure, but genome-wide analyses using DNA microarray or whole genome sequencing technology will give a broad view of the genome of HP in the near future. Factors that contribute to carcinogenesis include those that enable the bacteria to effectively colonize the gastric mucosa, those that incite a more aggressive host immune response, and those that affect host cell growth signaling pathways. Motility toward epithelial cells of the stomach is a vital feature of HP survival tactics. This is ensured by several factors. Spiraling movement is mediated by the FLA and FLAB proteins, which are designed to navigate the thick gastric mucus. Additionally, HP produces HP1069, a putative collagenase, which modifies the extracellular matrix and mucus layer, thus decreasing viscosity and allowing bacterial penetration. 75,76. In addition, HP expresses a variety of genes that contribute to buffering of stomach acid in order to maintain a relatively neutral pH. This includes a urease gene cluster consisting of 7 genes, of which urea/ure-B complex, comprising the urease enzyme, codes for 10% of the protein of HP and is vital for its survival. The CAG pathogenicity island is approximately 40 kb and contains 31 genes. The terminal gene of this island, CAG-A, is often used as a marker for the entire CAG locus. Compared with CAG-negative, CAG-A, strains, CAG-positive, CAG-A+, strains are associated with more severe inflammation, higher degrees of atrophy, and a greater chance of progressing to gastric adenocarcinoma. 7780. The estimated relative risk has ranged from 2 to as high as 28.4.34. However, many of the genes adjacent to CAG-A code for a type 4 secretion system, TFSS, often viewed as a molecule needle that injects bacterial proteins, e.g., CAG-A, into host cells. The remarkable finding that CAG-A is injected into host cells, where it is phosphorylated by SRC and CABL kinases, has raised the possibility that CAG-A could directly promote growth, migration, and transformation. Indeed, transgenic expression of HP CAG-A induces both GI and hematopoietic neoplasms in mice. 81 Other genes. Chapter 54 Adenocarcinoma of the stomach and other gastric tumors. 825-54 within the pathogenicity island are also believed to be important for disease, CAG or PIC-B, CAG-G, CAG-H, CAG-I, CAG-L, CAG-M, because they appear to be required for in vitro epithelial cell cytokine release, although they do not seem to have as great an effect on immune cell cytokine activation. 82-84 These Findings may explain the attenuated inflammatory response and lower cancer risk with CAG-A strains in vivo. 8588 Intracellular phosphorylation of CAG-A occurs at certain glutamate proline isoleucine tyrosine alanine (EPIA) motifs. 4 distinct EPIA motifs are described: EPIYAA, B, C, and D, whose prevalence varies by geographical region. 89,90 The motifs further influence the CAG-A-induced immune response as well as the related cancer risk. The odds ratio for gastric cancer is close to 7.3 in the case of one EPIYAC segment and can be up to 51 in case of 2 or more segments. 91,92 Also, genetic variations in further CAG-PI-related genes have been demonstrated to be associated with gastric cancer. 93 All strains of HP carry the VACA gene, which codes for a pore-forming, vacuolating toxin, but expression differs according to allelic variation. Approximately 50% of HP strains express the VACA protein, which has been shown to be a very powerful inhibitor of T-cell activation in vitro. 94 Although CAG-A map to different loci within the HP genome, the VACA protein is commonly expressed in CAG-A and strains. There are various forms of VACA, and the S1M1 strains are highly toxigenic. Other bacterial virulence factors, such as CAGE, may play a role in the modulation of apoptosis and the host inflammatory response, thereby contributing to disease manifestations. Indeed, virulent strains, CAG-A+, CAGE+, and VACA plus S1M1, appear to be more potent inducers of pro-inflammatory mediators than non-virulent strains, CAG-A, CAGE, and VACA, possibly explaining the higher association of CAG-A and strains with gastric cancer. 95 Dietary risk factors. Numerous dietary factors have been implicated as risk factors for gastric cancer. The decline in gastric cancer rates has coincided with the widespread use of refrigeration and the concomitant higher intake of fresh fruits and vegetables and lower intake of pickled and salted foods. Use of refrigeration for more than 10 to 20 years has been associated with a decreased risk of gastric cancer. 18 Lower temperatures reduce the rate of bacterial, fungal, and other contaminants of fresh food, as well as the bacterial formation of nitrites. Additionally, high intake of highly preserved foods may be associated with increased gastric cancer risk. 96 Potentially due to higher contents of salt, nitrates, and polycyclic aromatic amines. Much attention has been given to the effects of high nitrate intake. When nitrates are reduced to nitrite by bacteria or macrophages, they can react with other nitrogenated substances to form N-nitroso compounds that are known mitogens and carcinogens. In rats, N-nitroso compounds have been shown to cause gastric cancer. However, studies trying to link N-nitroso exposure to gastric cancer risk have been inconclusive, perhaps reflecting the fact that nitrate intake does not necessarily correlate with nitrosation levels. 97 A Swedish cohort study found a nearly twofold increased risk of gastric cancer associated with high dietary nitrate intake. 96 However, separate large cohort studies from Europe did not demonstrate an association between nitrate intake and risk of gastric cancer. 98,99 Another factor implicated in the development of gastric cancer is a diet high in salt, pickled foods, soy sauce, dried and salted fish, and meat. High salt intake has been associated with higher rates of atrophic gastritis in humans and animals in the setting of Helicobacter infection and increases the mutagenicity of nitrosated food in animal models. 18,100 High salt diets are associated with a roughly a 1.5 to 2-fold increased risk of gastric cancer. 101 Cohort and case control studies have also found an increased risk of gastric cancer associated with processed meat intake. 96,102 Possible mechanisms include higher bacterial loads, upregulation of HP-CAG-A expression, and increased cell proliferation and P21 expression. 100,103,104 There is a clear interaction between HP infection and dietary risk factors for gastric cancer, leading to a disproportional risk increase for HP-positive subjects with high intake of red or processed meat compared to individuals in whom only one of these risk factors is present. 105 Epidemiologic studies have had inconsistent findings with regard to fruit and vegetable consumption and risk. of gastric cancer. 106-109 Other foods or dietary factors that have been implicated as potential risk factors for gastric cancer are high intake of fried food, foods high in fat, high intake of red meat, and aflatoxins. 102-110-112 Diets with a high intake of fresh fish and antioxidants may be protective, also see later. 111-113-115 However, there are insufficient data to make definitive conclusions regarding these factors. Cigarette smoking. Tobacco has long been established as a carcinogen, and numerous epidemiologic studies have demonstrated an association between cigarette smoking and gastric cancer. 116 Several large cohort studies from Europe and Asia have reported a significantly increased risk of gastric cancer among smokers. 117-119 A recent meta-analysis found that, compared to never smokers, current smokers had a 1.5 to 2-fold increased risk of gastric cancer, both for the cardia and non-cardia region. 120 The authors also reported an increased association with greater amounts of smoking. Moist snuff is a smokeless tobacco product promoted as an alternative to cigarettes that has reportedly reduced levels of carcinogenic nitrosamines. Nevertheless, results of a Swedish cohort study demonstrated a 1.4-fold increased risk of non-cardia gastric cancer among regular snuff users. 121 Snuff exposure also increases the rate of gastric carcinogenesis in HP-infected mice. 122 Alcohol. Most epidemiologic studies have failed to demonstrate an association between alcohol consumption and cardia or non-cardia gastric cancer. 119-123-124 However, several meta-analyses suggest a small but significant association between heavy alcohol use and gastric cancer risk, RR, 1.16 to 1.87 in selected subgroups. 125-128 Although some of these analyses suggest that the risk is higher for junctional cancers than more distal gastric cancer, some authors state the opposite effect. Overall, the risk increase seems to be moderate and influenced by multiple factors, including tobacco consumption and physical activity. Interestingly, alcohol intake may increase the risk of gastric cancer in patients with certain polymorphisms of the alcohol dehydrogenase gene. 129 Obesity is a recognized risk factor for numerous GI malignancies. 130 Increased BMI is associated with a mild to moderate increased risk of gastric cardia cancer, but not non-cardia cancer. 131-134 Results of the National Institutes of Health American Association of Retired Persons, NIHAARP, Diet and Health Cohort Study demonstrated that morbid obesity, defined as a BMI greater than or equal to 35, as well as large waist circumference were associated with a 2 to 3-fold increased risk of gastric cardia cancer, but not non-cardia cancer. 135 A separate cohort study from the Netherlands also found an increased risk of cardia cancer with increasing BMI. 131 In a recent analysis of the EPIC cohort data from 391,456 individuals with 124 incident esophageal and gastric adenocarcinomas, neither 193 cardia cancers nor 224 non-cardia gastric cancers were associated with BMI. 136 The possible association between obesity and cardia cancer risk is likely mediated by pro-inflammatory cytokines and adipokines produced by intra-abdominal visceral fat. 137 826 Part 6 Stomach and duodenum. Genetic factors. As is true for most malignancies, both genetic and environmental factors play important roles in the pathogenesis of gastric cancer. Generally, intestinal-type gastric cancer is considered largely due to environmental causes, i.e., HP infection, whereas diffuse gastric cancer is considered a primarily genetic malignancy. In the case of intestinal-type gastric cancer, however, assigning relative values to environmental and genetic contributions is complex, given that the major environmental factor, HP, also tends to exhibit familial clustering. Nevertheless, in the future, gastric cancer types might rather be classified by genetic alterations and grouped to molecular subgroups with distinct carcinogenic mechanisms as well as clinical behavior, than to a histologic phenotype. 138 Overall, 10% of cases of gastric cancer appear to exhibit familial clustering 139, and family history is likely an independent risk factor even after controlling for HP status. 140,141 in a cohort study of relatives of patients with gastric cancer, siblings had a twofold increased risk of gastric cancer, adjusted for HP infection. 142 in a case control study from Japan. A positive family history was associated with a significantly increased odds of gastric cancer in women, or 5.10, but not in men. 143 a study from Scandinavia showed that having a twin with gastric cancer conferred a markedly higher relative risk for the disease, RR, 9.9 for monozygotic twins and 6.6 for dizygotic twins, leading the researchers to calculate that heritable factors accounted for 28% of gastric cancers, compared with 10% for shared environmental factors and 62% for non-shared environmental factors. 144 a recent meta-analysis of 32 studies including more than 80,000 individuals reported an increased pooled relative risk of 2.35, 95% CI, 1.96 to 2.81, for subjects with a positive family history of gastric cancer. The risk was even higher, RR 2.71, 95% CI, 2.08 to 3.53, if first-degree relatives had a gastric cancer diagnosis. 145 some of the familial clustering seen with intestinal-type gastric cancer may be related to genetic factors that play a role in the host immune response to HP infection. Data from South Korea indicate that individuals with a family history of gastric cancer more frequently have both HP infection and associated atrophic gastritis or intestinal metaplasia. 146 in a case control study from Scotland, relatives of patients with gastric cancer had a higher prevalence of atrophy and hypochlorhydria, but a similar prevalence of HP infection, compared with controls. 147 the greater prevalence of atrophy was confined to those patients with HP infection, suggesting the possibility these individuals were perhaps exhibiting a more vigorous immune response to HP. In a number of model systems, the development of gastric atrophy has been linked to a strong Th1 immune response. 50, 55, 148 thus, it was postulated that candidate disease susceptibility genes for gastric atrophy and cancer might be genes that participate in the innate and adaptive immune responses to HP infection. Inflammation is modulated by an array of pro- and anti-inflammatory cytokines, and several genetic polymorphisms have been described that influence cytokine response. With the recently started next-generation sequencing approaches, we may be able to determine whether families with increased gastric cancer incidence have a genetic predisposition for a more carcinogenic immune response. One such factor is IL-1 beta, an important pro-inflammatory cytokine and a powerful inhibitor of acid secretion. Indeed, there is an association between pro-inflammatory IL-1 gene cluster polymorphisms, IL-1B encoding IL-1 beta, and IL-1RN encoding its naturally occurring receptor antagonist, IL-1RA, and neoplastic progression in the setting of HP infection. Individuals with the IL-1 beta-31C or -511T and IL-1RN2/2 genotypes were shown in the study to be at higher risk for developing HP-dependent hypochlorhydria and gastric cancer. 149 The increased risk of progression to cancer with these genotypes was in the 2 to 3-fold range compared with non-inflammatory genotypes. The initial report was confirmed in other studies. 150-154 Subsequently, Wang and colleagues 155 demonstrated that carriers of the IL-1B511T/T genotype or the IL-1RN*2 allele had higher mucosal IL-1 beta levels than non-carriers, and also confirmed the association between the -511T/T genotype and severe gastric inflammation and atrophy. The importance of IL-1 beta carcinogenesis has now been demonstrated in a transgenic study, where stomach-specific expression of human IL-1 beta in transgenic mice led to spontaneous gastric inflammation and cancer that correlated with early recruitment of myeloid-derived suppressor cells to the stomach. 156 Of note, in a mouse model of Barrett esophagus and esophageal and EGJ tumors, IL-1 beta expression in the esophageal squamous epithelium also led to esophagitis and expansion of cardia stem cells. Forming gastroesophageal tumors, supporting the hypothesis that Barrett's-associated adenocarcinoma arises from the gastric cardia resembling a gastric cancer phenotype. 10 Additional associations with gastric cancer risk have been reported for genetic polymorphisms in TNF-alpha and IL-10. Pro-inflammatory genotypes of TNF-alpha and IL-10 were each associated with a twofold higher risk of non-cardiogastric cancer. When combined with pro-inflammatory genotypes of IL-1B and IL-1RN, patients with 3 or 4 high-risk genotypes showed a 27-fold greater risk of gastric cancer. 157 Additional studies have shown that polymorphisms of the toll-like receptor 4, TLR4, gene also increases the risk of gastric cancer. Carriers of the TLR4 plus 896G polymorphism had an 11-fold increased odds ratio for hypochlorhydria, and significantly more severe gastric atrophy and inflammation. 158 Accumulated evidence suggests that the genetic predisposition to gastric cancer may be largely determined by the TLR and cytokine responses to chronic Helicobacter infection. Polymorphisms in the TLR1 seem to protect against gastric cancer development, or 0.4, 95% CI, 0.22 to 0.72, and are furthermore related to alterations of downstream cytokine signaling. 159,160 The best described form of hereditary gastric cancer is the diffuse type gastric cancer that is seen in the presence of a germline mutation in the gene CDH1, which encodes the cell adhesion molecule E-cadherin. A large New Zealand kindred was found to have a germline mutation in the E-cadherin gene, and similar mutations have been reported in several additional kindreds, all with diffuse-type gastric cancer. 161-164 The age of onset of gastric cancer in individuals with CDH1 mutations is less than 40 years but can be highly variable, and the estimated lifetime risk of gastric cancer is close to 70%. 165-166 Germline CDH1 mutations are also associated with familial lobular breast cancer. 167-168 Small part of the familial clustering of gastric cancer can be attributed to other cancer syndromes. Patients with familial adenomatous polyposis have a prevalence of gastric adenomas ranging from 35% to 100%, and their risk of gastric cancer is close to 10-fold higher than that of the general population. 169 Dysplastic lesions in this specific group of patients frequently arise from fundic gland polyps, video 54.2, and develop at an early age. 170-171 Fundic gland polyps do not otherwise tend to progress toward dysplasia. Patients with hereditary non-polyposis colorectal cancer, HNPCC, syndrome have an approximately 11% risk of developing gastric cancer, predominantly of the intestinal type, with a mean age at diagnosis of 56 years. 172 Patients with juvenile polyposis also have a 12% to 20% incidence of gastric cancer. 173-174 In addition to the germline genetic alterations described earlier, next-generation sequencing techniques such as exome sequencing have led to the detection of new molecules and mechanisms that are involved in gastric carcinogenesis. In 8% to 10% of the gastric cancer patients, somatic mutations were identified in the ARID1A gene, also called BAF250A, SMARCF1, or OSA1, an accessory subunit of the SWISNF chromatin remodeling complex that is involved in processes of DNA repair, differentiation, and development. 175-178 Notably, cancers with EBV infection showed mutations of ARID1A in 73% of the cases. Chapter 54 Adenocarcinoma of the stomach and other gastric tumors 827-54 Additionally, ARID1A mutations were negatively associated with mutations in TP53 and occurred together with PIK3CA mutations. Patients with ARID1A alterations had longer recurrence-free survival, suggesting that these cancers belong to a molecular subgroup with distinct carcinogenic mechanisms as well as clinical behavior. 175-178 Analysis of somatic copy number aberrations have additionally shown significantly amplified genes, including therapeutically targetable kinases such as ERBB2, FGFR1, FGFR2, EGFR, and MET in gastric and gastroesophageal cancers. Tumor genetics. Although atrophy and intestinal metaplasia correlate with gastric cancer risk, direct cell progression through these stages has not been conclusively shown. Indeed, gastric cancer most likely arises from stem or progenitor cells present within the gastric mucosa rather than directly from terminally differentiated metaplastic cells. Investigators have for several decades sought to unravel the mutations responsible for gastric cancer initiation and progression to uncover a logical progression of acquired mutations akin to what is seen in colorectal cancer. However, gastric cancer does not follow a pattern like colorectal carcinoma progression. There is no clear-cut linear sequence of mutations in gastric cancers, and there is an even greater heterogeneity in genetic alterations. Although initial studies on large high-throughput data focused mainly on transcriptome analysis 179-181, the advent of more advanced genomic sequencing enables genome-wide analyses of the mutational landscape of gastric cancer. 182-184 The combined efforts of multiple research groups in international consortia enabled a more comprehensive integrative analysis of multilevel omics data from large cohorts. 9,13,138 TCGA consortium presented comprehensive data on 5 different platforms for about 300 gastric cancers. They demonstrated a good correlation of clustering between different levels of genomic data as well as epigenetic changes and transcriptome and even proteome analyses. 138 Based on their cluster analysis, the authors suggested a 4-group classification of gastric cancers, with the first group, EBV, being related to EBV infection, showing a dominant epigenetic hypermethylation profile, EBV. The second group, MSI, was positive for microsatellite instability, MSI, similar to the MSI subgroup of colorectal cancers. The remaining tumors were divided into a group, GS, with a low mutation rate and low frequency for copy number aberrations, called the genomically stable subtype, and a group, CIN, with high mutation rates and further related genomic changes, called the chromosomally unstable type. It is of note the GS group comprised predominantly diffuse-type tumors, whereas CIN tumors represented more intestinal-type cancers. There were some differences in the distribution of these subtypes with regard to location, with CIN tumors showing higher proportions with more proximal location, which was later also confirmed for esophageal adenocarcinomas. 9 The clinical relevance of this classification was suggested by the pattern of dominant changes in genes encoding for signaling pathways utilized in targeted treatment approaches. The Asian Cancer Research Group followed a similar approach in their cohort of 3,000 gastric cancers, although putting more focus on transcriptome data than the TCGA did. 13 Thus, Christisco et al. Presented a similar 4-group classification, also reporting properties as prognostic predictors for their groups. This classification was validated in several independent cohorts, including the group of TCGA patients. The actual genomic changes that have been described in these comprehensive studies are in line with results that have been published previously. Aneuploidy is common in gastric cancer, 60% to 75%, but cytogenetic studies have failed to identify any consistent chromosomal abnormality. Comparative genomic hybridization studies have shown that chromosome arms 4q, 5q, 9p, 17p, and 18q exhibit frequent decreases in DNA copy number, whereas chromosomes 8q, 17q, and 20q often have increased DNA copy number. 185 There is a general consensus that TP53 is the most commonly mutated gene in gastric cancer, 60% to 70% of gastric cancers, and that mutations in RAS, APC, and MYC are rare. 186,187 Loss of heterozygosity at the APC locus occurs more commonly. Another genetic abnormality found at high frequency, 60%, is the deletion or suppression of the fragile histidine triad gene, FHIT, a tumor suppressor locus on chromosome 3p. Genes that inhibit entry into the cell cycle, such as P16 and P27, show diminished expression in nearly one-half of gastric cancers. 188-193 Absence of P27 expression is associated with a poorer prognosis. 188-190 Absence of P16 expression is seen most commonly in poorly differentiated carcinomas but has no measurable impact on prognosis. 194 Diminished expression of P16 and P27 occurs in the absence of detectable mutations and is believed to be secondary to hypermethylation. 192 Many of these cancers show hypermethylation of a number of promoter regions, including the MLH1 promoter region, and show the high-level microsatellite instability, MSIH, phenotype, see Chapter 1. Multiple tumor suppressor genes have been shown to be methylated in gastric cancers. Emerging evidence suggests that these epigenetic changes, including global hypomethylation and promoter hypermethylation, occurs quite early in gastric carcinogenesis. In addition, it appears that DNA methylation changes also occur in the tumor-associated stromal fibroblasts, suggesting an important role for the tumor microenvironment. Overexpressions or amplifications of a number of growth factor pathways has been described, including COX-2, 70%, hepatocyte growth factor/scatter factor, HGF/SF, 60%, vascular endothelial growth factor, VEGF, 50%, c-Met, 50%, amplified in breast cancer 1, IB1, 40%, and beta-catenin, 25%. Table 54.2.195 Approximately 15% of gastric cancers have been reported to overexpress both EGF and EGF receptor, EGFR, consistent with an autocrine mechanism. Mutations in PIC3A, a gene that codes for a catalytic subunit of phosphatidylinositol 3 kinase, PI3K, has been found in up to 25% of gastric cancers analyzed. 196 In addition, mutations in genes encoding human protein tyrosine phosphatases, PTPs, were found by the same laboratory in 17% of gastric cancers, with the protein tyrosinase phosphatase receptor type the most frequently altered. 197 Gastric-specific tumor suppressor genes TFF1, trefoil factor 1, and RUNX3, runt-related transcription factor 3, which have now been identified and may represent gatekeepers of the gastric cancer pathway, are logical targets for further study. 198-199 Loss of TFF1 has been described in around 50% of gastric carcinomas, and TFF1 knockout mice develop spontaneous gastric enteral tumors. Mutations of TFF1 have also been described, and these enhance gastric cancer cell invasion through signaling pathways that include PI3 kinase and phospholipase C. 200 TFF1 expression is repressed by STAT3, and activation of STAT3 is also emerging as a key pathway that leads to gastric cancer. 54 RUNX3 most likely suppresses gastric epithelial growth by inducing P21 and BIM, attenuates WNT signaling, and is altered in 82% of gastric cancers. 201 Investigations into these genes and their contributions to the gastric cancer phenotype will prove valuable to our understanding of disease progression. MSI in dinucleotide repeats secondary to defects in DNA mismatch repair genes, such as MLH1 and MLH2, MUT-L homologs 1 and 2, have been mainly implicated in the development of colorectal cancer, and in particular the HNPCC syndrome. Patients with HNPCC have an 11% incidence of gastric cancer, suggesting that MSI may also play a role in the development of gastric cancer. 172 MSI is found in 15% to 50% of sporadic gastric cancers, with a higher prevalence in intestinal type of cancers. 202-207 Low-level microsatellite activity, e.g., MSI low. Can 828 Part 6 stomach and duodenum be found in 40% of areas of intestinal metaplasia in patients with gastric cancer. 207 and in 14% to 20% of adenomatous polyps. 205-207-208 MSIH occurs in only 10% to 16% of gastric cancers. MSI is associated with the less frequent occurrence of TP53 mutations, well to moderately well differentiated histology, and distal location in the stomach. Studies that have examined the effect of MSI on patient survival have shown inconsistent results. 208,209 When the findings are taken together, it would appear that MSI does play a role in the pathogenesis of gastric cancer, likely before the development of intestinal metaplasia, see Fig 54.3, and is most commonly due to methylation of the MLH1 promoter. There are increasing data regarding the genetics of diffuse-type gastric cancer. 138,184 Several families with hereditary diffuse gastric cancer have been found to carry a germline mutation in the E-cadherin gene, CDH1, 161, 163,210, 211. However, mutations in CDH1 have also been described as a dominant feature in sporadic diffuse-type gastric cancer. Further evidence supporting a role for E-cadherin in the pathogenesis of gastric cancer comes from studies showing that suppression of E-cadherin expression occurs in 51% of gastric cancers, with a higher percentage found in diffuse-type cancers. 212 Furthermore, E-cadherin underexpression is associated with higher rates of lymph node metastases and reduced survival. 213, 214 The overall rates of CDH1 mutations in gastric cancer are low. Thus, the decreased expression of E-cadherin seen in gastric cancer is likely secondary to hypermethylation of the CDH1 promoter, which occurs in 50% of gastric cancers and 83% of diffuse-type gastric cancers. 215 E-cadherin is a transmembrane protein that connects to the actin cytoskeleton through alpha and beta catenins to establish cell polarity and mediates homophilic cellular interactions. 216, 217 Decreased expression of E-cadherin is believed to promote dissociation of cancer cells from their cell matrix, enhancing the migration and invasion of gastric cancer cells. Expression of alpha-catenin is also decreased or absent in 68% of gastric cancers. 218 Therefore, E-cadherin appears to act as a tumor suppressor gene that may be important in the pathogenesis of diffuse gastric cancer. Other alterations that commonly occur in diffuse-type gastric carcinoma are alterations in unrelated genes as well as changes in the RAS homolog gene family, member A gene, RHOA, which seems to be exclusive to this histological subtype. 184 Perhaps as important as the genetic alterations acquired during the progression to gastric adenocarcinoma is the question, in what target cells do these changes occur? For a cell to accumulate the quantity of genetic changes necessary for autonomous growth, it must be long-lived. For these reasons, the current thinking is that a resident tissue stem cell is the target of genetic mutations and becomes the cancer stem cell capable of autonomous growth and with metastatic potential. Recently, several elegant genetic lineage tracing studies in mice established markers that allow the distinction of two different types of GI stem cells. Crypt-base columnar cells, CBC, are fast-cycling stem cells expressing LGR5 and CD133, PROM1. 219,220 A villain transgene has allowed the identification of a multipotent progenitor located in the lower third of a subset of antral gastric glands. 221 Whereas multiple intestinal stem cell markers could also be identified in the antrum. Interestingly, LGR5 shows lineage labeling in some antral gastric glands and in the gastric cardia. 220 Slower cycling cells, which are usually found at the +4 position of the crypts of the antrum, i.e., the fourth epithelial cell in the crypt, counting from the bottom of the crypt upward, are characterized by a pronounced expression of BMI1 and TERT. 222, 223 Although these two types of cells are functionally interconnected. 224 Their exact hierarchical relationship remains to be identified. Seigel and colleagues demonstrated a direct KAGA-dependent activation of LGR5 positive gastric stem cells, thus reporting a further mechanism by which H. pylori infection induces gastric carcinogenesis. 225 The same group also suggested orchestration of epithelial hyperproliferation and gland hyperplasia as a response to the infection by cells of the stromal compartment, mainly myofibroblasts involving HP-induced WNT signaling. 226 In the gastric oxyntic glands, the proliferative zone with the gastric stem cell has been localized to the isthmus, the middle portion of the tubule, and cells are thought to migrate bidirectionally to supply gastric surface mucus cells that coat the gastric pits, and gastric parietal and zymogenic cells that comprise the base of the gland. 227 The gastric corpus stem cell has not yet been identified, none of the markers discussed earlier labels any specific cells within the gastric isthmus. Recently, progenitor cells, e.g., KRT19+ and TFF2+ cells, have been shown through lineage tracing studies to label different gastric progenitor cells. 228,229 Typically, columnar metaplasia is positive for TFF2 and KRT19. Given that intestinal metaplasia arises in the gastric mucosa and in the esophagus, it is plausible that a similar stem cell gives rise to both. Regardless of their localization, CBC or +4 position, or their function, GI stem cells depend on signals from the stem cell niche, such as pericryptal myofibroblasts and neighboring differentiated epithelia. 230 Important signaling pathways required for stem cell maintenance and proliferation comprise the WNT, Notch, bone morphogenetic proteins, and hedgehog pathways. 231 There is increasing knowledge on the interaction of the local stromal microenvironment with the epithelium. Table 54.2 Genetic abnormalities in gastric adenocarcinoma. Abnormalities: approximate gene frequency, %. Microsatellite instability 15 to 50, DNA aneuploidy 60 to 75, deletion/suppression P53 60 to 70, FHIT, fragile histidine triad gene, 60 APC, adenomatous polyposis coli gene, loss of heterozygosity 50, DCC, deleted in colorectal cancer gene, loss of heterozygosity 50, decreased expression due to hypermethylation P1650, TFF1, human trefoil factor 1 gene, 50 P2750, MLH1, human MUT-L homolog 1 gene, 15 to 20 E-cadherin 50, amplification/overexpression COX270 HGF, hepatocyte growth factor, 60. VEGF, vascular endothelial growth factor, 50 c-MET 50 IB1, amplified in breast cancer 1, 40 beta-catenin 25 EGFR, EGF receptor gene, 15 mutations PI3K, phosphatidylinositol 3 kinase gene, 25 PTPRT, protein tyrosine phosphatase receptor type gene, 17 Chapter 54 Adenocarcinoma of the stomach and other gastric tumors 829 54 Regulator of the biological behavior of gastric cancer cells are cancer-associated fibroblasts, that have been shown to modify TGF-beta-dependent signaling, increasing cellular motility, and, therefore, invasiveness. 232,233 The density of tumor-infiltrating lymphocytes and their vicinity also modifies the tumor's aggressiveness and therefore has an impact on the prognostic outcome in patients with adenocarcinomas in the stomach or at the EGJ. 234,235 This factor is partly reflected by the level of systemic inflammation. 236 Systemic inflammation can be partly mediated by the visceral adipose tissue, and it has been demonstrated that omental adipocytes enhance the invasiveness of gastric cancer by activation of PI3K anite signaling in the tumor cells. 237 Besides the valuable data from in vitro and ex vivo models, approaches for virtual microdissection of next-generation high-throughput and sequencing data are promising to enhance our understanding of the network of different cellular components and its impact on tumor initiation, promotion, and progression, including invasive and metastasizing behavior. Fig. 54.4. Premalignant conditions: chronic atrophic gastritis. Chronic atrophic gastritis, which is defined as the loss of specialized glandular tissue in its appropriate region of the stomach, is an established morphologic change that occurs along the sequence toward the development of gastric cancer. 238 The presence of atrophic gastritis has an annual incidence of progression to gastric cancer of approximately 0.1% to 1.0%. 239-243 The extent of atrophic gastritis within the stomach correlates with risk of progression to cancer. 244-246 And large studies have demonstrated that only patients with extensive mucosal atrophic changes at baseline develop gastric adenocarcinoma during long-term follow-up. 247 There are two forms of atrophic gastritis, see Chapter 52. The more common is environmental multifocal atrophic gastritis, which is associated with HP infection and more likely to be associated with metaplasia. The presence of HP infection is associated with an approximately 10-fold increased risk of atrophic gastritis. 248 There is considerable regional variation in the prevalence intestinal gastric metabolic proliferative mesenchymal MSI-EBV H. pylori nutrients immune response bile microenvironment MSS/EMT MSS/TP53 plus CIN implications on treatment options, diagnostic markers, prognostic markers classic histopathology transcript profiling genome profiling / multilevel data integration GSMSI-EBV MSS/TP53 diffuse Fig 54.4 molecular stratification of gastric cancer. Displayed is an overview of recent concepts of gastric cancer subtyping. These classifications emerged from data gathered by transcript profiling high-throughput study as well as next-generation sequencing and transcend the classic histopathological classification, left. Early transcriptome data suggested dichotomous groups that showed similar features to the classic Lauren types, middle, top, before more functional interpretation was enabled by pathway analyses, middle, bottom. Recent concepts are usually based on multilevel data integration, but still originate mainly from genome data like the TCGA approach, EBV, MSI, microsatellite instability positive cancer, GS, genomically stable cancer, CIN, chromosomal instability cancer, or from transcriptome data like the Asian Cancer Research Group, ACRG, approach, MSI, microsatellite instability positive cancer, MSS/EMT, microsatellite stable with transcript signature for epithelial mesenchymal transition, MSS/TP53+. Microsatellite stable with TP53 mutation, MSS/TP53, microsatellite stable with wild-type TP53. A special interest lies in the understanding of factors that drive each phenotype, mostly the interaction with the tumor microenvironment. 830 Part 6 Stomach and duodenum of atrophic gastritis in HP-infected individuals, with a roughly threefold increase in Asia compared to Western countries. 248,249 The second form of atrophic gastritis, autoimmune metaplastic atrophic gastritis, is associated with antiparietal cell and intrinsic factor antibodies. This form of atrophy is confined to the body and fundus. Autoimmune metaplastic atrophic gastritis is associated with pernicious anemia and an increased gastric cancer risk, albeit not as high as that seen with HP-induced MAG, owing most likely to a lesser degree of inflammation. 240,250 Mechanisms underlying the increased risk of gastric cancer in the setting of gastric atrophy may be related to low acid output, hypo or achlorhydria, which predisposes to increased bacterial overgrowth with non-Helicobacter organisms, greater formation of N-nitroso compounds, and diminished ascorbate secretion into the gastric lumen. 251 Additionally, circulating gastrin levels increase in response to the reduced acid output. Gastrin is a known growth factor for gastric mucosal cells, and sustained elevations of gastrin may contribute to abnormal growth and increased risk of neoplastic progression. 252,253 Intestinal metaplasia and dysplasia. Intestinal metaplasia, IM, can be subdivided into three categories, as classified by Philippe's group. 254 Type I, complete, IM contains goblet cells that secrete sialamucins and mature, non-secretory absorptive cells. Type 1 IM is not a risk factor for gastric cancer. Type 2, incomplete, IM contains few if any absorptive cells, columnar intermediate cells in various stages of differentiation secreting neutral or acidic sialamucins, and goblet cells secreting sialamucins and / or occasionally sulfomucins. Type 3, incomplete, is less differentiated than Type 2, with the intermediate cells secreting mainly sulfomucins and the goblet cells secreting sialo and / or sulfomucins. Type 2 or 3 IM is associated with an approximately 20-fold increased risk of gastric cancer. 255,256 Early gastric cancer develops in 42% of patients with Type 3 IM within 5 years of follow-up, suggesting that IM represents a precursor lesion for the intestinal form of gastric cancer. 256 A large nationwide cohort study from the Netherlands on 92,250 individuals identified a 0.25% annual progression rate toward cancer in patients with gastric IM. 243 However, whether cancer arises from areas of IM or whether IM simply represents a marker for higher gastric cancer risk remains unclear, mainly due to the focal and patchy appearance of this condition. As is the case with atrophic gastritis, the prevalence of IM in HP-infected individuals is higher in Asia, 40%, as compared to the West. 248,249 Although it is generally accepted that IM is associated with an increased risk for the intestinal type gastric cancer, patients with diffuse type tumors also show a high prevalence of IM in the non-tumorous gastric mucosa. 257,258 As mentioned earlier, a number of recent studies have revealed that intestinal metaplasia is not the only possible metaplastic precursor of gastric cancer. Although controversy exists as to the sequence and connection of mucosal lineage changes associated with increased risk for gastric cancer, there is a general agreement that the loss of acid-secreting parietal cells, also known as oxyntic atrophy, is a prerequisite for induction of metaplasia. Antralization of the fundus, or the presence of metaplastic glands in the fundus with a general phenotype similar to that of the antral or pyloric glands, also known as pseudopyloric metaplasia, is frequently associated with intestinal-type adenocarcinoma. This phenotype has also been called spasmolytic polypeptide-expressing metaplasia, SPEM, 259 and is characterized by the presence of trefoil factor 2, TFF2, or spasmolytic polypeptide, immunoreactive cells in the gastric fundus, with morphologic characteristics resembling those of deep antral gland cells. SPEM was observed in association with over 90% of resected gastric cancers in 3 studies in the USA, Japan, and Iceland. 259-261 Data from mice demonstrate that Helicobacter-induced expression of TFF2 is associated with CD44 plus cancer stem cell in the same compartment of the gastric mucosa, therefore possibly facilitating further malignant progression. SPEM and intestinal metaplasia might share equal importance as putative paraneoplastic lesions in the stomach. Nevertheless, it remains to be determined whether either or both these metaplasias can progress to dysplasia or neoplasia. Alternatively, intestinal metaplasia may potentially reflect a further benign attempt by the mucosa to increase repair in the face of chronic infection and inflammation. Histologic assessment of gastric dysplasia and adenocarcinoma is based on the Vienna classification, the result of an international consensus conference of GI pathologists in 2000, Table 54.3.262. The prevalence of gastric dysplasia ranges from as low as 0.5% in low-risk areas 263 to 20% in high-risk areas. 264 Prospective studies have shown that low-grade dysplasia may regress in up to 60% of cases, whereas 10% to 20% progress to high-grade dysplasia. Fig. 54.5.265-267 High-grade dysplasia rarely regresses, and is associated with a 2% to 6% annual incidence of progression to gastric cancer. 267,268 In a prospective cohort study from the Netherlands, the presence of high-grade dysplasia was associated with a 40-fold Table 54.3 Padova International Classification System for Gastric Dysplasia Category Definition Histologic Description I Normal gastric architecture with absent or minimal inflammatory infiltrates reactive foveolar hyperplasia. The general architecture is well preserved, with evidence of hyperproliferative epithelium, enlarged nuclei, and mitotic figures. Intestinal metaplasia type I closely resembles the morphology of the small intestine, with absorptive enterocytes, well-defined brush borders, and well-formed goblet cells. Type 2. Incomplete metaplasia with irregular mucus vacuoles, absence of brush borders, and difficult to identify absorptive enterocytes. Cells secrete mainly sialamucins. Type 3. Same as type 2, except cells secrete mainly sulfomucins. 2 Indefinite for dysplasia unable to discern whether cells are neoplastic or non-neoplastic, usually found in the setting of inadequate biopsy specimens and presence of architectural distortion and nuclear atypia. 3. Non-invasive neoplasia. Phenotypically neoplastic epithelium that is confined to glandular structures inside the basement membrane, includes adenomas. Should be divided into low-grade and high-grade. 4. Suspicious. 4. Invasive cancer. Presence of neoplastic epithelium where invasion cannot be clearly identified. V. Invasive cancer. Invasive carcinoma adapted from Ruggie M., Correa P., Dixon M., et.al. Gastric dysplasia, the Padova International Classification. AMJSURG Pathol 2000, 24:167-76. Chapter 54 Adenocarcinoma of the stomach and other gastric tumors. 831-54 Increased risk of progression to gastric cancer. High-grade dysplasia is often associated with synchronous cancer and can be uni or multifocal. 269 Gastric polyps. The prevalence of gastric polyps in the general population is approximately 0.8% to 2.4%. 270,271 Gastric polyps consist predominantly of fundic gland polyps, 50%, hyperplastic polyps, 40%, and adenomatous polyps, 10%. 271,272 The clinical course of fundic gland polyps is generally benign, and they are detected with increasing frequency in the era of PPI use. In a series of 599 consecutive patients who underwent upper endoscopy, use of PPIs for more than 5 years was associated with a nearly 4-fold increased risk of fundic gland polyps. 273 The rate of malignant transformation of these polyps is generally quite low, 1%. And confined to polyps larger than 1 cm. 274 One notable exception to the benign nature of fundic gland polyps is in familial adenomatous polyposis, video 54.3. In this group, the prevalence of fundic gland polyps ranges from 51% to 88%, with dysplasia present in over 40% of cases. 170,171 Hyperplastic polyps are generally benign, often multiple, and are typically observed in the setting of chronic inflammatory conditions, e.g., chronic atrophic gastritis, pernicious anemia, chronic enteral gastritis, adjacent to ulcers and erosions, and especially at sites of gastroenterostomies. Over time, the polyps may regress, remain stable, or increase in size, and they often regress following HP eradication. Men and women are equally affected, and the polyps typically appear in mid to late adult life. 275 The rare hyperplastic polyps that undergo malignant transformation often have areas of dysplasia or intestinal metaplasia and typically form a well-differentiated intestinal type cancer. 274 In contrast to other polyps of the stomach, gastric adenomas undergo malignant transformation at a high rate. When gastric adenomas were followed by serial endoscopy with biopsy, progression through dysplasia to carcinoma in situ developed within 4 years in approximately 11% of cases. 276 Endoscopic biopsy of gastric polyps can be associated with significant sampling error. 277 The British Society of Gastroenterology published guidelines in 2010 regarding the management of gastric polyps. 278 Among the recommendations were: 1. All gastric polyps should be at least biopsied. 2. All gastric adenomas, symptomatic polyps, and polyps with dysplasia should be removed, and 3. HP, if present, should be eradicated in patients with hyperplastic or adenomatous polyps. Decisions regarding surveillance intervals should be made on an individual basis. Previous gastrectomy. It has been reported by several groups that gastric surgery for benign conditions can predispose patients to a higher risk of gastric cancer, beginning 20 years after the surgery. 279-282 The risk is greatest for those who underwent surgery before the age of 50 years, perhaps reflecting the long lag period necessary between the operation and the development of cancer. 281 The cancers tend to occur at or near the surgical anastomosis on the gastric side, only rarely do they reside on the intestinal side of the anastomosis. 283 Numerous theories have been proposed to explain the increased propensity for cancer to form at the surgical anastomosis site. They include hypochlorhydria resulting in bacterial overgrowth with increased production of nitrites, chronic enterogastric reflux of bile salts and pancreatic enzymes, which are potent gastric irritants, and atrophy of the remaining fundic mucosa due to low levels of antral hormones, including gastrin. 18,284,285. The Bilroad 2 operation with gastrojejunostomy predisposes to the development of cancer at a 4-fold higher rate than a Bilroad I procedure with gastroduodenostomy, suggesting that bile reflux may be a significant predisposing factor. 280 HP and associated intestinal metaplasia are found less frequently in post-gastrectomy gastric cancers as compared to distal gastric cancers in the non-operative stomach. 286 It is unclear whether screening for gastric cancer in this population of patients in areas of low cancer incidence would be cost-effective. With the advent of HP eradication therapy as well as PPIs, the number of gastric resections for peptic ulcer disease has decreased dramatically, significantly reducing the impact of the post-gastrectomy state as a risk factor for gastric cancer. PUD, see also Chapter 53. Large epidemiologic studies have demonstrated a consistently increased risk of gastric cancer in patients with a history of a gastric ulcer. In a cohort study, Swedish adults who were followed for an average of 9 years, a history of gastric ulcer was associated with a 1.8-fold increased risk of gastric cancer. 287 Interestingly, a history of duodenal ulcer was associated with a reduced risk of gastric cancer. These findings were replicated in a case control study of U.S. veterans. 288 The associations were Fig. 54.5 Histopathology of gastric dysplasia. Left. Low-grade dysplasia is characterized by a proliferation of neoplastic epithelial cells with nuclear pseudostratification and hyperchromasia in the absence of architectural changes. Right. High-grade dysplasia has more severe cytologic abnormalities with abnormal architectural features, including irregular fused or cribriform glands and papillae. H&E stains. 832 Part 6 Stomach and duodenum confined to non-cardiogastric cancer. There was no association between history of gastric ulcer and cardiac cancer. 288 It is unclear whether gastric ulcers per se predispose to the development of cancer. The increased risk may be mediated by infection with HP, which can lead to atrophic gastritis, intestinal metaplasia, and cancer. Ménétrier disease, see also Chapter 52, in a review of case reports. 15% of patients with Ménétrier disease had associated gastric cancer. 289 Including several cases that documented a progression from dysplasia to cancer. 290,291 Because of the rarity of Ménétrier disease, it has been difficult to study its relationship with gastric cancer in any controlled fashion, and no recommendations regarding endoscopic surveillance can be made. Screening and surveillance. The majority of the literature regarding screening for gastric cancer comes from East Asia, where the prevalence of this disease is among the highest in the world. 292 Since 1960, the Japanese have been performing mass screening using upper GI barium studies followed by endoscopy if any suspicious lesions are found, and this continues to represent the recommended approach based on current Japanese cancer screening guidelines. 293 Japanese researchers have reported a sensitivity of 66% to 90% and a specificity of 77% to 90% for this screening method. 294 However, survey studies have shown that, in clinical practice, upper endoscopy is the most widely used screening test for gastric cancer in Asia. 295 Not surprisingly, studies from Japan have also shown that screening results in diagnosis of gastric cancer at earlier stages, with one study reporting more than half of screened cases diagnosed as stage I. 296 Long-term follow-up data from the Japanese Public Health Center cohort showed that subjects who underwent screening had a nearly 50% reduced risk of death from gastric cancer. 297 A separate cohort study from Japan found a 25% to 35% risk reduction in death from gastric cancer among those who participated in gastric cancer screening. 298 However, similar risk reductions were seen for death from all causes, casting a level of uncertainty on the true magnitude of benefit associated with screening with respect to preventing death from gastric cancer. The serum pepsinogen (PG) test is increasingly used to screen for patients at highest risk for having perineoplastic gastric lesions. The stomach produces two types of pepsinogens, PGI and PGII. In chronic atrophic gastritis, production of PGI is reduced due to reduction of the number of gastric chief cells, whereas PGI levels remain relatively constant or can sometimes even be raised in response to inflammation, see Chapter 52. Therefore, both low serum PGI levels, 70 mg/L, and a low PGI/2 ratio, 3.0, are useful for the identification of patients with atrophic gastritis. 292 Large prospective cohort studies have shown that baseline PGI, PGI/2, and HP antibody levels combined can successfully identify patients at highest risk for developing gastric cancer for whom referral to endoscopy might be appropriate. 299 302 Screening with upper endoscopy is likely to be cost-effective in moderate to high-risk populations, such as older Asian men. 303 However, in populations with a lower incidence of gastric cancer, screening is less likely to have the same degree of beneficial impact. European consensus guidelines recommend endoscopic surveillance in patients in whom extensive atrophy of the gastric mucosa has been diagnosed. 304 Gastroscopy is recommended every 3 years in these patients. Despite initial analyses that such an approach can be cost-effective, prospective randomized trials are currently underway to support this strategy. 305,306 A USA recent meta-analysis suggests that this strategy can only be cost-effective in low-incidence Western countries when applied to selected individuals with further risk factors. 307 Prevention. Given the lethal nature of gastric cancer and its link to chronic infection and inflammation, much attention has been paid to the possibility of chemoprevention of gastric neoplastic lesions. The approach most studied has been HP eradication, but consideration has also been given to supplementation with antioxidants and the use of NSAIDs and COX-2 inhibitors. Eradication of HP. Eradicating HP leads to a decrease of the subsequent risk of gastric cancer. There is little question that chronic inflammation in a variety of organ systems can lead to malignancy and that HP eradication can reduce or alleviate gastric inflammation. HP eradication can lead to decreased oxidative stress and cell proliferation. 308 In addition, limited studies involving eradication of gastric Helicobacter organisms in Mongolian gerbils suggest that eradication of infection can partially reverse atrophy and metaplasia and inhibit progression to gastric cancer. 309 Studies in mice confirm the reversibility of metaplasia and prevention of gastric cancer with early eradication. With later eradication, cancer progression was slowed and cancer mortality dramatically decreased. 19 Nevertheless, definite proof of the cancer-preventing effect of HP eradication by prospective randomized trials in humans is lacking. This is partly due to the need for high numbers of at-risk patients to be included to achieve adequate power with regard to the rare endpoint of incident gastric cancer, and partly due to ethical concerns to randomize patients into a study arm that would leave them untreated with a Class I carcinogen. One approach to tackle the first issue has been to examine the effect of HP eradication on premalignant conditions, such as gastric atrophy and intestinal metaplasia. Thus, a majority of studies has shown a beneficial effect in preventing progression of gastric disease. 310-314 In a randomized, placebo-controlled trial from China of 587 patients with HP infection, assignment to eradication was associated with a significantly reduced risk of progression of intestinal metaplasia, odds ratio, 0.63, 0.314. In contrast, a randomized placebo-controlled trial of Mexican adults did not demonstrate a benefit of HP eradication for the prevention of histologic progression. 313 A prospective, randomized placebo-controlled trial in 1,630 healthy HP-positive individuals sought to determine whether HP eradication in a high-risk population in China would reduce the incidence of gastric cancer. Although no overall group benefit was seen in the group receiving HP eradication, there was a reduction in gastric cancer incidence in the subgroup of patients who did not already have precancerous lesions, gastric atrophy, intestinal metaplasia, or dysplasia, at study initiation. It is possible that some of the patients in the eradication arm had passed the earlier mentioned point of no return, when cellular alterations had sufficiently accumulated to promote cancer. 315 Recent meta-analysis of randomized trials found that HP eradication is associated with a significant 35% reduction in the risk of gastric cancer. 22 There may even be benefit to HP eradication after treatment of early gastric cancer in light of the high rate of associated multifocal dysplasia. In an open-label randomized controlled trial of patients with resected early gastric cancer by Fukase et al., HP eradication was associated with a reduction in the risk of development of metachronous gastric cancer, odds ratio, 0.35, 95% CI, 0.0161 to 0.775. Although there are conflicting results, recent meta-analyses support the initial findings of Fukase and colleagues. 316,317 Chapter 54 Adenocarcinoma of the stomach and other gastric tumors 833-54 In Western countries, gastric cancer prevention has not been extensively pursued due to the lower prevalence of HP infection and decreasing incidence of gastric cancer. However, a cost-effectiveness model by Parzon-Net and colleagues 318 suggested that screening and treatment of HP infection would be potentially cost-effective in the prevention of gastric cancer, particularly in high-risk populations, if it was assumed that treatment of HP infection prevented 30% of attributable gastric cancers. Using a more conservative 10% reduction in gastric cancer risk, an analysis from the United Kingdom also concluded that HP eradication was cost-effective. 319 An alternative option would be population-based vaccination against HP infection. But development of an effective vaccine as well as definition of the right time point for vaccination have demonstrated to be difficult. Despite a recent study showing promising results, it is not clear yet if a clinically applicable vaccine will be available in the near future. 320 Aspirin and other NSAIDs, including COX-2 inhibitors among other effects. Aspirin and other NSAIDs inhibit cyclooxygenases. COX-1 is constitutively expressed in the GI tract. COX-2 expression is generally not observed in normal GI mucosa, but is induced in multiple epithelial malignancies, including gastric cancer. 321,322 COX-2 expression is associated with aggressive cell growth in both human and mouse models of cancer. 323-326 and has been found to be overexpressed in 70% of gastric cancers. 327 In this setting, COX-2 could potentially promote the growth of tumors, inhibit apoptosis, and increase angiogenesis. COX-2 expression has been reported to be elevated in paraneoplastic lesions, including both intestinal metaplasia and dysplasia, and COX-2 expression appears to diminish after HP eradication. 328 Multiple epidemiologic studies have demonstrated a consistent association between NSAID use and reduced risk of gastric cancer. 329-332 In a case control study from Los Angeles County, NSAID use for more than 5 years was associated with a reduced risk of non-cardiac cancer, odds ratio, 0.61. And there was a significant NSAID dose-related effect. 330 A nested case control study using the General Practitioners Research Database in the United Kingdom found that long-term users of non-aspirin NSAIDs had a decreased risk of gastric cancer, odds ratio, 0.65, although there was no effect of aspirin use on the risk of gastric cancer. 332 This study was in contrast to a recent study in 2 UK cohorts from England and Scotland on 3,833 patients with gastric cancer and 4,654 patients with esophageal cancer. 333 In these cohorts, long-term aspirin use was not associated with cancer-specific mortality after diagnosis of gastric cancer, pooled adjusted HR, 1.06, 95% CI, 0.85 to 1.32. On the other hand, recent meta-analyses reported a significant association between any NSAID use and reduced risk of gastric cancer, RR, 0.78, 95% CI, 0.72 to 0.85, with broadly similar findings for both acetylsalicylic acid aspirin, ASA, and non-ASA NSAIDs and a slightly more pronounced effect for non-cardiac cancer. 334 In a randomized controlled trial of HP-negative patients with intestinal metaplasia, there was no difference in the rate of regression of intestinal metaplasia. After 2 years between patients receiving the COX-2 selective inhibitor rofecoxib and placebo. 335 This trial was limited by the relatively short follow-up period and use of premalignant endpoints. In a separate randomized controlled trial in patients with HP and histology showing chronic atrophic gastritis, or worse, both HP eradication and 24 months of the COX-2 selective inhibitor selecoxib resulted in histologic regression, although no additive effect was observed. 336 In a summary analysis of randomized trials of aspirin versus no aspirin for various outcomes, those studies with 10 to 20 years follow-up reported a reduced risk of gastric cancer in those assigned to aspirin. Odds ratio 0.42. 337 Further trials in high-risk patients are warranted to determine if NSAIDs are effective for gastric cancer prevention. Statins, the widely prescribed class of HMG-CoA inhibiting cholesterol-lowering drugs, have been found in numerous epidemiologic studies to be associated with decreased risks of various malignancies. In addition to their cholesterol-lowering properties, statins also have anti-proliferative and pro-apoptotic effects. 338 A population-based case control study from Taiwan found a significantly reduced risk of gastric cancer in patients prescribed statins, odds ratio 0.68, with greater risk reduction observed among those with the highest cumulative statin use. 339 In a separate case control study of diabetics from South Korea, a history of statin use was associated with an 80% reduced likelihood of gastric cancer. 340 A pharmacy database study from the Netherlands found a significant association between statin use and a decreased risk of cancer of any type. However, there was no significant association with gastric cancer, although the number of cases was relatively small. 341 Meta-analyses agree on an overall favorable profile of statins with regards to gastric cancer risk, showing a risk reduction that ranges between 32% to 44%. 342,343 Interestingly, there was no statistically significant difference between Western or Asian cohorts with regards to this effect. Future randomized controlled trials of various statins in patients at high risk for gastric cancer will help define the role of this class of drugs as chemopreventive agents. Antioxidants. Chronic inflammatory states such as HP gastritis can result in the generation of free radicals derived from oxygen and nitrogen. 344 These free radicals can promote carcinogenesis via numerous different means, including direct DNA damage and inhibition of DNA repair mechanisms, inhibition of apoptosis, and activation of cellular proliferation pathways. Antioxidants such as carotenoids and vitamin C and E bind with reactive oxygen and nitrogen species to neutralize their damaging effects. Epidemiologic data support a relationship between increased antioxidant intake and reduced risk of gastric cancer. 345-349 In a nested case control study from Japan, low plasma beta-carotene levels were associated with an increased risk of gastric cancer. 347 A case control study from Korea found that elevated nitrate/antioxidant intake ratios were associated with increased risk of gastric cancer. 348 In a Swedish cohort study, high levels of vitamin A, retinol, and alpha and beta-carotene intake were associated with a 50% risk reduction in gastric cancer. 349 A recent meta-analysis on the impact of alpha and beta-carotene intake analyzed data from 13 case control and 8 cohort studies and reported inconsistency of the respective results between case control and cohort studies. 350 Randomized controlled trials have shown inconsistent effects of antioxidant supplementation on gastric cancer risk. In a randomized placebo-controlled trial of antioxidants, either vitamin A, C, or E, in patients with precancerous gastric lesions, nonatrophic or atrophic gastritis, intestinal metaplasia or dysplasia, antioxidant supplementation did not result in either reduced histologic progression or increased histologic regression. 351 A randomized controlled trial in China also found no effect of combined vitamin C, E, and selenium supplementation on the prevalence of a combined endpoint of atrophic gastritis, intestinal metaplasia, dysplasia, or cancer. 352 In a 10-year follow-up of the General Population Nutrition Intervention Trial in China, subjects who received a combination of selenium, vitamin E, and beta-carotene were found to have reduced mortality from gastric cancer. 353 Interestingly, Lee et al. Reported in a systematic analysis 834 Part 6 stomach and duodenum of the data available in 2013 that although high dietary intake of vitamin C, vitamin E, and alpha and beta-carotene resulted in a reduced risk for gastric cancer, the actual blood levels of these factors were not associated with gastric cancer risk. 354 Given a lack of convincing chemopreventive effect as well as the results of the beta-carotene and retinol efficacy trial, in which subjects who received beta-carotene and vitamin A had an increased risk of lung cancer. 355 Antioxidant supplementation for the prevention of gastric cancer cannot yet be recommended. This is in line with a recent Cochrane meta-analysis on the effect of selenium supplementation on cancer prevention. 356 Although there are some data from observational studies suggesting a minor decrease in the site-specific risk for stomach cancer, studies are heterogeneous and prospective randomized controlled trials would be needed to support this hypothesis. Other dietary factors. Green tea is widely consumed in Asian countries and is hypothesized to have protective effects against cancer of the upper digestive tract. Polyphenols and other metabolites present in green teas, such as epigallocatechin 3 gallate, EGCG, and other catechins, have a variety of anti-tumor effects, including induction of apoptosis, inhibition of tumor cell growth and proliferation, and reduction in COX-2 expression. 357-359 EGCG also has antioxidant properties and may have anti-inflammatory properties as well. 360-361 Although case control studies have shown an inverse association between the risk of gastric cancer and the consumption of green tea, cohort studies have largely failed to show an association. 362-363 One cohort study from Japan did report a reduced risk of gastric cancer in women with high green tea consumption, but no change in risk among men. 364 A recent meta-analysis of observational studies suggested a slightly decreased risk by long-term high-dose green tea intake. 365 But in the absence of prospective controlled trials, green tea cannot be recommended as chemoprevention for gastric cancer. Similar results are available on the effect of garlic consumption. 366 Adherence to a Mediterranean diet has the potential to reduce gastric cancer risk and related mortality by nearly 30%, RR 0.72, 95% CI, 0.60 to 0.86. 367 A recent model calculation predicted that increasing fruit and vegetable intake would prevent a relatively high proportion of gastric cancer cases by 2025, mostly in developing countries. 368 Clinical features. Early gastric cancers are asymptomatic in up to 80% of cases. When symptoms do occur, they tend to mimic peptic ulcer disease. With advanced gastric cancer, the most common symptoms are weight loss, 60% of patients, and abdominal pain, 50%. 369 Other presenting symptoms include nausea, vomiting, anorexia, dysphagia, melena, and early satiety. Pyloric outlet obstruction can occur with tumors of the antrum and pylorus, and tumors of the cardia can cause dysphagia due to involvement of the lower esophageal sphincter and development of pseudoachalasia. See Chapter 44. 370 Rarely, paraneoplastic syndromes occur. There have been reports of thrombophlebitis, Trousseau-Sign neuropathies, nephrotic syndrome, and DIC. 371-373 Dermatologic paraneoplastic syndromes are also uncommon and include hyperpigmented patches in the axilla, acanthosis nigricans, see Chapter 25, and the sudden onset of seborrheic dermatosis, senile warts, and pruritus, sign of Les Urtralat. 374 The physical exam is usually unremarkable. Cachexia and signs of bowel obstruction are the most common abnormal findings. Occasionally, it is possible to detect an epigastric mass, hepatomegaly, ascites, and lower extremity edema. 375 Laboratory studies are generally unrevealing until the cancer reaches advanced stages. Anemia and a positive test result for fecal occult blood may occur from chronic bleeding of an ulcerated mass. Hypoproteinemia can occur. Liver enzyme values, particularly serum alkaline phosphatase levels, can be elevated secondary to hepatic metastases. Gastric cancer is metastatic at the time of diagnosis in 33% of cases. 376 The most common sites of metastasis are the liver, 40%, and peritoneum. 377 Other sites of spread include periumbilical lymph nodes, Sister Joseph nodule, left supraclavicular sentinel nodes, Fierco node, the pouch of Douglas, rectal shelf of Bloomer, and the ovaries, Krukenberg tumor. Gastric cancer has also been reported to metastasize to the kidney, bladder, brain, bone, heart, thyroid, adrenal glands, and skin. 375 There are reports of unusual presentations of metastatic disease, such as shoulder-hand syndrome from bone metastasis, diplopia, and blindness from orbital and retinal metastases, and viralization due to Krukenberg tumors. 378 381 Diagnosis. Endoscopy EGD is currently the procedure of choice for the diagnosis of gastric cancer, Fig 54.6a. When a non-healing gastric ulcer is found, at least 6 to 8 biopsy specimens from the edge and base of the ulcer are recommended. 382 The American Gastroenterological Association has recommended that an upper endoscopy be performed in patients who are older than 55 years with new-onset dyspepsia and in patients younger than 55 years who have alarm symptoms, weight loss, recurrent vomiting, dysphagia, evidence of bleeding, anemia. 383 Dyspeptic patients in whom an empirical trial of PPIs and eradication of HP do not relieve symptoms should undergo prompt endoscopic evaluation as well. The basis for these recommendations is the low incidence of gastric cancer in individuals younger than 55 years. The yield of upper endoscopy for the detection of gastric cancer in patients with occult bleeding and a normal colonoscopy will vary, based on the patient's baseline risk of gastric cancer. In Japan and other areas of high gastric cancer prevalence, chromoendoscopy, magnification endoscopy, and narrowband imaging are used alone or in combination as aids in the detection of early gastric cancer, see Fig 54.6b. Distinct irregular mucosal surface and vascular patterns have been found to correlate with the presence of dysplasia and carcinoma. 384 There are also ongoing investigations into the utility of newer techniques such as autofluorescence and confocal microendoscopy for the diagnosis of early gastric neoplasia. 385,386 In the past, barium studies have been reported to have 60% to 70% sensitivity and 90% specificity for the detection of advanced gastric cancer. 387 Nevertheless, upper GI series has been largely replaced by upper endoscopy as the initial test of choice for the diagnosis of gastric cancer. A classification system has been developed for early gastric cancer based on endoscopic appearance. 388 The purpose of which is to assess early lesions for risk of submucosal invasion as well as risk of lymph node spread, Fig 54.7. The three types include superficial polypoid, type 0I, superficial flat/depressed, types 0I IAC, and superficial excavated, type 03, lesions. The most commonly observed subtype is 0IIC, the non-polypoid depressed lesion. 388 This classification system is used most often in Japan, where endoscopic mucosal resection and submucosal dissection are frequently performed for early gastric neoplasia. CT gastrography. Although CT colonography has gained significant attention for its potential role as a screening modality for colon polyps and colon cancer, CT gastrography has also been studied for the diagnosis of early gastric cancer. In a study from South Korea of 39 patients with early gastric cancer, CT gastrography had a Chapter 54 adenocarcinoma of the stomach and other gastric tumors. 835 54 sensitivity of 73% to 76% and good interobserver reliability, kappa equals 0.84. 389 Only small studies have been performed thus far using this imaging modality, and CT gastrography cannot yet be recommended for screening outside of the research setting. Serum markers. To date, no reliable serum marker has been identified with high sensitivity and specificity for the diagnosis of gastric cancer. Low serum PGI levels, low ratios of PGI to PGII, and hypergastrinemia have been reported in patients with atrophic gastritis and intestinal metaplasia, but the results for the detection of gastric cancer have been mixed. 390,391 In a study of 17,000 Japanese males, a positive PG test, defined as PGI 50 μg/L, and PGI/2 3.0, in combination with upper GI series, identified gastric cancer in only 0.28% of subjects. However, 88% of these cancers were early cancers. 392 Additionally, 89% of the cancers identified by the PG test alone were early gastric cancers. The major limitation of this test is the low specificity for the diagnosis of gastric cancer. 393 Therefore, assessment of serum PG is currently being used to identify patients with paraneoplastic conditions that benefit from further diagnostic testing, e.g., by endoscopy. Serum CEA and carbohydrate antigen, CA, 19-9 have both been extensively studied for the diagnosis of gastric cancer. The sensitivities of these markers are especially low for early gastric cancer. 394 And elevated levels are also seen in other epithelial malignancies. These tumor markers are frequently elevated in recurrent gastric cancer, especially in patients who had elevated levels prior to surgical resection. 395 The diagnostic quality of these markers, however, remains low, and they might instead have a role as prognostic indicators, because both CEA and CA19-9 are associated with unfavorable clinical pathological characteristics and poor outcome for gastric cancer patients. 396,397 Other studies have identified, among others, TGF-beta1, CA72-4, tumor M2 pyruvate kinase, and hepatocyte growth factor as potential markers. Gastric cancer. 398,401 But as for CEA and CA19-9, clinical use might be rather found as prognostic indicators, particularly for recurrence after surgical treatment. 402 Apart from classic serum markers, a recent focus has been placed on so-called liquid biopsies, mainly the assessment of circulating tumor cells and circulating tumor DNA. Recent advancements in enrichment and sequencing approaches enables the analysis of these factors, which can deliver information on mutational profile and tumor heterogeneity even without the needs for invasive tissue sampling. 403 Although data on diagnostic accuracy and value as prognostic marker are still inconsistent, the potential use to guide targeted therapy becomes more interesting. A prominent example is the assessment for HER2 amplification by analysis of circulating plasma DNA. 404,405 This approach has the potential to overcome problems of tissue analyses with tumor heterogeneity and sampling error. 406 Classification and staging. Several classification systems exist to further define gastric cancer and predict prognosis. As mentioned earlier, see Fig 54.2. Gastric cancers can be subdivided into intestinal and diffuse types. Gastric cancer can also be divided into early and advanced lesions. Early gastric cancer is defined as a cancer that does not AB Fig 54.6 Endoscopic examples of gastric cancer. A. Ulcerated gastric adenocarcinoma mass lesion. B. Chromoendoscopic view of a superficial depressed gastric cancer, highlighted with indigo carmine arrow. A. With permission from the Gastrolab Endoscopy Archives. The WASA Workgroup on Intestinal Disorders, 2008 accessed October 14, 2008, at http://www.gastrolab.net/pa-269.htm B, from Toyota H, Tanaka K, Hamada Y, et.al. Endoscopic diagnosis of hypopharyngeal, esophageal, and gastric neoplasm. Dig Endosc 2006, 18S41-3. 0 IP protruded, pedunculated superficial, elevated flat excavated superficial shallow, depressed protruded. Cecil 0 is 0 IIA 030 IIB 0 IIC Fig 54.7 Schematic representation of the major variants of type 0 neoplastic lesions of the stomach, polypoid, IP and is, non-polypoid, IIA, IIB and IIC, non-polypoid and excavated. 3. From the Paris Endoscopic Classification of Superficial Neoplastic Lesions, Esophagus, Stomach and Colon, November 30 to December 1, 2002. Gastrointestinal 2003, 58S3-43. 836 Part 6 Stomach and duodenum invade beyond the submucosa, regardless of lymph node involvement. This form of cancer has a much higher prevalence in the Far East, especially Japan, and carries a very favorable prognosis, with 5-year survival rates greater than 90% being reported in Asia and greater than 80% in Western countries. 407-410 The most commonly used clinical staging classification system for gastric cancer is the TNM system, used by the International Union Against Cancer and the American Joint Committee on Cancer. 411-412 In the TNM staging system, T, tumor, indicates the depth of penetration. Fig 54.8 T1A denotes a tumor that invades the lamina propria or mucosa, T1B denotes invasion of the submucosa, T2 denotes invasion of the muscularis propria, T3 denotes invasion of the subserosal connective tissue, T4A denotes invasion of the serosa, visceral peritoneum, and T4B denotes invasion into adjacent organs or structures. N, nodes, indicates the amount of lymph node invasion, N0 denotes no lymph node involvement, N1 denotes involvement of 1 to 2 lymph nodes, N2 denotes involvement of 3 to 6 lymph nodes, and N3 denotes involvement of 7 or more lymph nodes. M, metastasis, indicates the presence of metastases, with M0 denoting no metastases and M1 denoting distant metastases, including positive peritoneal cytology, Table 54.4. In the American Joint Committee on Cancer Staging Manual, cardiac cancer, tumors within 5 cm of and crossing the GE junction, is now classified together with esophageal and GE junction tumors. 412 Recent studies have investigated reclassification of gastric cancer based on biological characteristics. The prospect of incorporating tumor biology into staging classification systems is intriguing, although future validation studies are required for this to occur. Accurate staging in gastric cancer is important for treatment decisions. EUS is the best studied modality for the staging of gastric cancer and remains the test of choice for assessing tumor depth and nodal involvement. However, improvements in image quality for both CT and MRI make these studies potential alternatives and adjuncts to EUS. EUS allows the visualization of the 5 layers of the gastric wall. The superficial gastric mucosa is represented by an echogenic first layer, and the deeper mucosa by a hypochogenic second layer. The submucosa is represented by an echogenic third layer, the muscularis propria as a hypochogenic fourth layer, and the serosa as an echogenic fifth layer. EUS can also identify and biopsy submucosal lesions, such as gastric lymphomas and stromal tumors. These lesions typically involve thickening of the submucosa and muscularis propria and may appear as gastric fold thickening on barium studies or endoscopy. Based on results of a meta-analysis of EUS for gastric cancer staging, EUS has sensitivity of 86% and specificity of 91% to distinguish T1-2 versus T3-4 tumors. 413 Intramucosal lesions, T1A, are identified with 83% sensitivity and 79% specificity. EUS may be particularly useful for identifying early gastric cancer lesions amenable to endoscopic mucosal resection or submucosal dissection. 414, Fig 54.9. In terms of end staging, the rate of detection of perigastric nodes with EUS is comparable to staging with CT. 415,416 EUS is slightly less accurate in the assessment of nodal status as compared to depth of tumor invasion, with 69% sensitivity and 84% specificity to distinguish positive from negative lymph node status. 413 A particular difficulty with end staging lies in the fact that many small lymph nodes can also harbor metastases, and thus understaging can occur. CT and PET advances in imaging technology have greatly improved the ability of CT to stage gastric tumors. Although not as extensively studied as EUS, multi-detector row CT, MDCT, by which the wall of the stomach can be seen as 3 layers, an inner layer corresponding to the mucosa, an intermediate layer corresponding early gastric cancer, advanced gastric cancer, T1, 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 T2, T3, T2, T3, T4, T4 EUS appearance: mucosa, superficial, mucosa, deep, submucosa, muscularis propria, serosa, adjacent organ, tis hyperechoic, 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 hypoechoic, hypoechoic. Fig 54.8. Classification of gastric adenocarcinoma by depth of invasion, T classification. In the TNM classification, T denotes depth of invasion, TIS designates carcinoma in situ, T1 tumors are confined to the mucosa, T1A, and submucosa, T1B, T2 tumors invade the muscularis propria but not the serosa, T3 tumors penetrate the subserosal connective tissue without involving the visceral peritoneum or contiguous structures, and T4 tumors invade the serosa, visceral peritoneum, and may involve adjacent organs and tissues. In early gastric cancer, the disease is confined to the mucosa and submucosa, T1, regardless of nodal involvement. Table 54.4 Clinical staging of gastric cancer based on the TNM classification N0, N1, N2, N3, M1, NEN, TIS 0 T1 IAIB IIA IIB 4 T2 IB IIA IIB 3A 4 T3 IIA IIB 3A 3B 4 T4A IIB 3A 3B 3C 4 T4 B 3B 3B 3C 3C 4 is, in situ, M, metastases, N, node involvement, T, tumor. Asterisk from Brearley J.D., Gaspadero is M.K., Witkin C., editors. TNM classification of malignant tumors. 8th ed. Hoboken, New Jersey, Wiley Blackwell, 2017. Chapter 54 Adenocarcinoma of the stomach and other gastric tumors 837-54 to the submucosa, and an outer layer of slightly higher attenuation corresponding to the muscularis propria and serosa, appears to have comparable accuracy to EUS in terms of both T and N staging. The loss of fat planes between the gastric mass and an adjacent organ suggests tumor invasion. The accuracy of MDCT for overall T staging ranges from 77% to 91%, and discriminates serosal involvement with an accuracy of 83% to 100%. 417,418 accuracy with respect to N staging may be as high as 89% with MDCT. 419,420 As with all other imaging modalities, CT has difficulty discerning metastases in lymph nodes smaller than 5 mm. At present, the role of CT is mainly for the detection of distant metastases and as a complement to EUS for assessing regional lymph node involvement. It is not yet clear whether EUS or MDCT, or the combination, is superior for T and N staging in gastric cancer, and the underlying technology continues to evolve and improve. PET scanning alone is not recommended as a sole imaging test for gastric cancer staging, largely because most gastric adenocarcinomas have low F18-fluorodeoxyglucose, FDG, uptake and there are false positives as well, see Chapter 52.421. However, in patients initially staged as having localized gastric cancer, combined PET/CT increases the detection of metastatic disease by 10%, thus altering clinical management. 422 Laparoscopy with peritoneal lavage. Approximately half of gastric cancer patients with metastatic disease have cancer involving the peritoneum. 377 Current imaging techniques such as EUS and CT have limited ability to detect peritoneal dissemination. In fact, up to one-third of patients with seemingly resectable disease will have evidence of peritoneal spread at the time of staging laparoscopy. 423 National Comprehensive Cancer Network guidelines recommend consideration of laparoscopy with peritoneal lavage for patients with seemingly resectable disease in whom neoadjuvant chemotherapy is being considered. 424 However, the USA has been slow to adopt this practice. A study using SEER Medicare data found that only 8% of gastric cancer patients who had any surgery underwent laparoscopy. 425 Other imaging modalities. MRI with gadolinium has also been used for gastric cancer staging. It is similar to CT in its advantages, ability to find distant metastases, and weaknesses, need for adequate gastric distension. The accuracy of MRI ranges from 90% to 93% for T staging and from 91% to 100% for N staging. 417 However, given the small number of studies, MRI cannot yet be advocated as the test of choice for staging gastric cancer. Rest aging after neoadjuvant treatment. The accuracy of rest aging gastric cancer after neoadjuvant chemotherapy decreases considerably. EUS has less than 50% accuracy for both T and N rest aging, and similarly disappointing results have been reported for post-treatment staging with CT.426. However, the use of preoperative clinical staging to assess response to neoadjuvant chemoradiation may correlate well with both overall and disease-free survival. 427 Rest aging is therefore primarily used to rule out distant metastasis as part of the assessment for surgical resectability. Prognosis and treatment. Overall, the 5-year survival rate in the USA from gastric cancer is 27%, compared with 64% for colon cancer. Point 4 The TNM classification is used to stratify disease into 4 clinical stages, I through 4, to predict prognosis in patients treated with gastrectomy, see Table 54.4. The survival data from Japanese studies are generally superior to those seen in Western countries, perhaps because of the preference in Japan for extended lymphadenectomy or because of less understaging than is found in Western countries. 428 There are data to suggest that large tumor size, greater than 5 cm, may be independently associated with worse survival, independent of nodal status or overall tumor stage. 429 Surgical resection remains the primary curative treatment for gastric cancer. However, survival after surgery alone is poor, 20% to 50% at 5 years, necessitating efforts to improve the outcomes for this group of patients using perioperative chemotherapy or postoperative, adjuvant, chemoradiotherapy. In addition, surgical resection often provides the most effective palliation of symptoms, particularly those of obstruction. In some cases, surgery is required for diagnosis, as in cases of non-healing gastric ulcers with negative biopsy results and persistent pyloric outlet. AB Fig 54.9 Gastric cancer staging. A. Endoscopic image of an early gastric cancer showing a 25 mm protruding mass located on the posterior wall of the antrum. B. EUS image of the lesion, showing the hypoechoic mucosal mass, arrow, with an intact submucosal layer. From Kim J.H., Song K.S., Yun Yat, ETAL. Clinicopathologic factors influence accurate endosonographic assessment for early gastric cancer. Gastrointestinal Disc 2007, 66:901-8. 838 Part 6 Stomach and duodenum obstruction suggesting an antral carcinoma. Surgery should be attempted in most cases of gastric cancer. However, in the presence of extensive involvement of diffuse type cancer, or linitis plastica, bulky metastatic disease, retroperitoneal invasion, or peritoneal carcinomatosis, or if the patient has severe comorbid illnesses, the prognosis may be sufficiently poor to make the value of resection questionable. Surgery, and laparoscopy in particular, can be useful in the staging of cancer. Laparoscopy can help identify primary tumor resectability, peritoneal deposits, and appropriate candidates for neoadjuvant therapy. Laparoscopic peritoneal lavage has been used to detect intraperitoneal free cancer cells. A positive peritoneal lavage correlates significantly with eventual development of overt peritoneal metastases. 430 In general, total gastrectomy is performed for proximal gastric tumors and for diffuse gastric cancer, and partial gastrectomy is reserved for tumors in the distal stomach. Large, randomized multi-center trials in France and Italy comparing subtotal with total gastrectomy for adenocarcinoma of the antrum found no differences in 5-year survival rates or operative mortality. 431,432 Some centers have argued for performing a complete splenectomy with gastrectomy. However, several retrospective and prospective studies found that concurrent splenectomy increased morbidity and had either no effect on or worsened survival. 433,434 The extent of lymphadenectomy accompanying the gastrectomy has been a subject of debate for many years. The Japanese advocate a more extensive lymph node dissection, D2 resection, than their Western counterparts, D1 resection, and have higher published survival rates. A D2 resection entails resection of the nodes of the celiac axis and the hepatoduodenal ligament in addition to the perigastric lymph nodes taken in a D1 procedure. The differences in reported survival rates may reflect the fact that the Japanese have a much higher incidence of early gastric cancer, and the more extensive lymph node dissection performed in Japan may find more positive lymph nodes, making survival rates of Japanese patients with N0 staging appear to be higher than those of their potentially understaged Western counterparts. A large multi-center randomized trial from the Netherlands reported no significant improvement in 5-year survival and more postoperative deaths and complications with D2 lymphadenectomy than with the more conservative D1 lymphadenectomy. 435 In a subsequent 15-year follow-up of these patients, the investigators reported no significant difference in overall survival, although there was significantly reduced gastric cancer-related mortality in the D2 resection arm, 37% versus 48% in the D1 arm. 436 A British randomized trial of 400 patients likewise showed no benefit from more extensive surgery. 5-year survival rates were 35% for D1 resection and 33% for D2 resection. 433 At present, data are insufficient to support extended lymph node resection in centers outside Japan. To prevent understaging, the current recommendation is a minimum D1 lymphadenectomy with removal of at least 15 nodes. 437 Endoscopic mucosal resection and submucosal dissection. Advances in endoscopic techniques have permitted endoscopic mucosal resection, EMR, and endoscopic submucosal dissection, ESD, to be used as curative therapies for select early gastric cancers, EGCs. Video 54.4. This technique has been used widely for intestinal-type cancers in Japan and South Korea, where studies have shown that only 3.5% of patients with EGCs smaller than 2 to 3 cm have lymph node involvement, making these lesions amenable to local therapy. Lesions larger than 4.5 cm have a greater than 50% chance of spread into the submucosa, are associated with positive nodes, and are therefore less likely to be endoscopically resectable. 438 The following criteria have been suggested for EMR in gastric cancer. 1. The cancer is located in the mucosa and the lymph nodes are not involved, as indicated by EUS examination. 2. The maximum size of the tumor is less than 2 cm when the lesion is slightly elevated, type IIA, and less than 1 cm. When the tumor is flat or slightly depressed, type IIB or IIC, without an ulcer scar. 3. There is no evidence of multiple gastric cancers or simultaneous abdominal cancers, and 4. The cancer is of the intestinal type. 439 Despite these guidelines, it is generally not possible to remove lesions larger than 1.5 to 2.0 cm en bloc by EMR, and piecemeal removal of EGC is associated with decreased rates of curative resection. 440 ESD is a technique developed in Japan and permits en bloc resection of larger EGCs, as well as selected tumors with submucosal invasion. With ESD, submucosal injection is performed, followed by use of endoscopic electrosurgical knives to resect the entire tumor. Fig 54.10. 441 Endoscopic prediction of EGCT stage is less accurate than in the colon and lesion margins are less distinct. Also, unlike in the colon, lymphatics are found within gastric mucosa and gastric lesions do not follow the adenocarcinoma sequence of transformation. As a result of these differences, en bloc resection by ESD affords the best chance of accurate histological staging and potential cure for EGC. In addition to an R0 resection, ESD allows for more precise histopathologic assessment of depth of invasion and lymphovascular involvement and permits appropriate assessment for risk of lymph node metastasis. If pre-procedure evaluation does not reveal regional lymph node involvement, much larger superficial lesions can be resected. The Japanese have developed expanded criteria for ESD for early gastric cancer. 1. Mucosal intestinal type cancer of any size without ulceration. 2. Mucosal intestinal type cancer less than 3 cm with ulceration, and 3. Submucosal intestinal type cancer less than 3 cm and with submucosal invasion less than 500 μm. 442,443 As experience with this ESD has increased, en bloc resection rates are now reported to be over 90%, with local recurrence rates lower than 3%. 441 Owing to the large size of some of the lesions being resected, the risk of gastric perforation is relatively high, 2% to 6%. 444,445 However, Perforations recognized early can generally be treated conservatively with closure using endoscopic clipping. 441 Western guidelines for gastric ESD include both absolute and expanded criteria depending on lesion size and morphology. Recent gastric ESD studies from Western centers have shown comparable outcomes to that of Eastern centers, particularly in critical endpoints such as R0 resection and complications. In a prospective observational study of 191 EGC resected by ESD from a single Western referral center and block, and R0 resection rates of 98.4% and 90.2%, for lesions meeting guideline criteria, and 89.0% and 73.6%, for lesions meeting expanded criteria, respectively, were seen. For patients with EGC but higher risk of lymph node involvement and who are poor candidates for surgical gastrectomy, combination ESD with laparoscopic lymph node dissection may be an alternative approach. 446 There are no published randomized clinical trials comparing surgery to endoscopic resection for early gastric cancer. Chemotherapy in Western countries. Approximately 75% of patients with gastric cancer have disease that has spread to the perigastric lymph nodes or have distant metastases at the time of diagnosis. 447 Patients presenting at early stages are often treated with surgery in combination with perioperative chemotherapy with curative intent. Numerous clinical trials have been performed evaluating the role of adjuvant chemotherapy after curative resection for gastric cancer. 448 The majority of the studies were inconclusive, but a series of meta-analyses of these trials suggested a 15% to 20% reduction in the risk of death in patients who received adjuvant. Chapter 54 Adenocarcinoma of the stomach and other gastric tumors. 839 54 Chemotherapy. 449, 450 Randomized trials of cisplatin or epirubicin-based adjuvant chemotherapy have largely failed to show a benefit. 451, 452 The optimal regimen for first-line chemotherapy has yet to be clearly established. Whether a 3-drug regimen is more effective than a potentially less toxic doublet is a point of controversy. Historically, clinical trials have included as a group esophageal, EGJ, and gastric adenocarcinomas. Pooling these conditions may have hidden relevant location-dependent outcomes, although Chow and co-workers demonstrated that, despite a slightly better outcome in esophageal and EGJ adenocarcinomas, no statistical differences were found in terms of overall survival or response rates in the three groups. 453 Nevertheless, the new characterization of esophageal and gastric cancer within the TCGA suggests that these tumors are quite different on molecular levels and should therefore be likely treated differently and be approached in at least different treatment trials. The European Organization for Research and Treatment of Cancer expert panel differentiated treatment and staging recommendations for tumors near the GE junction. Preoperative chemoradiation was recommended for adenocarcinoma of the esophagogastric junction, AEG, type I and II tumors. For AEG type 3, cardia, tumors, perioperative chemotherapy was suggested as the best choice. Neoadjuvant chemotherapy appears to benefit patients with resectable disease. In the UK MAGIC trial, 503 patients with gastric, gastroesophageal, or distal esophageal cancer were randomly assigned to undergo surgery alone or surgery following neoadjuvant epirubicin, cisplatin, and 5-fluorouracil, 5-FU. Compared to surgery alone, the neoadjuvant group had significantly improved 5-year, 36% versus 23%, progression-free, and overall survival. 454 As a result, preoperative, neoadjuvant, chemotherapy is now considered the standard treatment option prior to surgery for gastric cancer. In the recent FLOT4A10 trial, perioperative chemotherapy with docetaxel, oxaliplatin, and fluorouracil/leucovorin, FLOT, significantly improved progression-free survival and overall survival, OS, among patients with resectable gastric cancers compared with epirubicin, cisplatin, and fluorouracil or capecitabine, ECF/ECX, suggesting FLOT as a preferred regimen in perioperative treatment of patients with adenocarcinomas of the stomach or gastroesophageal junction. Chemoradiation. Combined chemoradiation after surgical resection appears to be effective at improving progression-free and overall survival in gastric cancer. The intergroup trial 0116 randomized 603 patients with gastric or gastroesophageal cancer to undergo surgery alone or surgery followed by 5-FU, leucovorin, and radiation therapy. Subjects in the surgery alone group had a shorter median survival time, 27 months versus 36 months, and a worse overall and relapse-free survival. 455 Following publication of the results of this study, adjuvant chemoradiation became the standard of care in the USA, although the optimal chemotherapy regimen is not yet clear. Early studies of the use of neoadjuvant chemoradiation as well as recent as yet unpublished trials have also shown promising results. 456 ADBECF Fig 54.10 Endoscopic submucosal dissection for early gastric cancer, EGC. A. Conventional endoscopic view showing an EGC type 0 IP at the angularis. B. Endoscopic view after indigo carmine spraying to enhance the lesion margin. C. Normal tissue surrounding the lesion has been marked with a needle knife. D. A circumferential cut around the marking dots has been made with an insulated tip electrosurgical knife. E. The base after en bloc submucosal dissection of the lesion has been performed. F. The specimen stretched and pinned on a wood plate before immersion in formalin. From Lee Il, Wu CS, Tung Sy, ETAL endoscopic submucosal dissection for early gastric cancers, experience from a new endoscopic center in Taiwan. J. Clin Gastroenterol 2008, 42:42-7. 840 Part 6 Stomach and duodenum intraperitoneal chemotherapy. Because systemic chemotherapy is largely ineffective for peritoneal metastasis, intraperitoneal, IP, chemotherapy can be considered in patients whose tumors are resected for cure but have a high likelihood of microscopic residual disease. In a randomized trial of 248 patients with gastric cancer, postoperative hyperthermic IP chemotherapy was associated with improved overall survival compared to surgery alone. 457 The treatment benefits were most pronounced in patients with stage 3 and 4 disease, serosal invasion, and lymph node metastases. Although a second clinical trial reported similar results, 458 other studies have failed to demonstrate a benefit of hyperthermic IP chemotherapy. 459,460 A meta-analysis of studies of IP chemotherapy for patients with resectable gastric cancer reported a significantly reduced risk of death in patients who received hyperthermic IP chemotherapy, odds ratio, 0.60, 461 At present, the use of hyperthermic IP chemotherapy should be confined to patients enrolled in clinical trials. According to data presented at the 2018 Gastrointestinal Cancers Symposium in San Francisco, undergoing cytoreductive surgery plus hyperthermic intraperitoneal chemotherapy in 180 patients treated in 19 French centers between 1989 and 2014 resulted in improved overall survival compared with resection alone for 97 patients with gastric cancer with peritoneal carcinomatosis. Unresectable disease. Unfortunately, up to one-third of patients with gastric cancer will have unresectable disease at the time of diagnosis. 376 Chemotherapy for locally advanced gastric cancer without distant metastases can result in shrinking of the tumor to the point where successful curative resection is possible. 462,463 Even when curative surgery is not possible, chemotherapy has been shown both to improve survival as well as quality of life compared to best supportive care in this group of patients. 464 Therefore, patients with inoperable locally advanced and/or metastatic, stage 4, disease should be considered for systemic treatment, chemotherapy, to improved survival and quality of life compared with best supportive care alone. A meta-analysis by Wagner and colleagues 465 demonstrated a small but significant survival benefit for combination chemotherapies, with a median survival of 8.3 months with combination regimens and 6.7 months for single-agent therapies. As expected, toxicity was increased in the combination schedules, and thus, combination chemotherapy should only be considered in patients with good performance status. Doublet combinations of platinum and fluoropyrimidines are generally used, and there remains controversy regarding the utility of triplet regimens. Although there is no single standard of care in advanced gastric cancer, there is some evidence coming from meta-analyses. Drugs related to increased survival in phase 3 trials are cisplatin, docetaxel, and trastuzumab, a monoclonal antibody that interferes with the HER2/neu receptor. HER2 is amplified and is a key driver of tumorigenesis in 7% to 34% of gastric cancers. In the TOGA phase 3 multi-center randomized study, patients with gastric cancer and HER2 overexpression received chemotherapy and trastuzumab, resulting in a median overall survival of 13.8 months, compared with 11.1 months in those treated with chemotherapy alone. 466 Further manipulation of this pathway using the novel anti-HER2 directed agents pertuzumab and T-DM1, in addition to dual EGFR/HER2 blockade with lapatinib, may yield positive results. Consequently, tumor assessment for HER2 overexpression should be performed, and the addition of trastuzumab to palliative chemotherapy should be considered for every patient with HER2 plus gastric adenocarcinoma, Fig. 54.11. In contrast, targeting of the epidermal growth factor receptor, EGFR, pathway in combination with chemotherapy in unselected patients has not been fruitful to date. Similarly, use of the anti-angiogenic monoclonal antibody bevacizumab was not successful in a large global randomized trial. 467-471 Careful selection of patient subsets will become a key factor in future clinical trials. Gastric cancer, adenocarcinoma, operable stage T1NO preferred pathway. Consider endoscopic/limited resection preoperative chemotherapy surgery adjuvant chemoradiotherapy HER2 negative, platinum and fluoropyrimidine-based doublet or triplet regimen HER2 positive, trastuzumab and CF/CX. Consider clinical trials of novel agents second-line postoperative chemotherapy surgery reassess palliative chemotherapy inoperable or metastatic best. Supportive care if unfit for treatment operable stage T1 no V38V49. Cancer, EMSO clinical practice guidelines for diagnosis, treatment, and follow-up. An-Oncle 2016, 27, Supple 5, Fig 54.11 Algorithm for the treatment of gastric cancer. Taken from Smith E.C., Verhage M., Alam W., ETAL Gastric Chapter 54 Adenocarcinoma of the stomach and other gastric tumors 841-54 Several randomized clinical trials have demonstrated efficacy of multi-drug cisplatin-based regimens. 472-473 A newer clinical trial using the EOX regimen, epirubicin, oxaliplatin, and capecitabine xylota, was found to be non-inferior to cisplatin-based regimens, and had a median survival of 11.2 months. 474 The benefit of the EOX regimen is the substitution of oxaliplatin and capecitabine for cisplatin and 5-FU, respectively, resulting in greater convenience, ease of administration, and potentially fewer side effects. Second-line chemotherapy may also be superior to best supportive care, but again no standard regimens have been defined. Monotherapy with docetaxel or irinotecan has been shown to be superior to best supportive care. 475-477 and a recent study showed no superiority of irinotecan over weekly paclitaxel. Thus, monotherapy with irinotecan or taxanes such as paclitaxel can be considered an option in advanced gastric cancer patients as a second-line treatment. 478 The anti-VEGFR2 monoclonal antibody ramucirumab has shown activity in 2 randomized phase 3 trials. As a single agent, it is associated with a survival benefit comparable to cytotoxic chemotherapy in the second-line setting, whereas ramucirumab in addition to paclitaxel is associated with a survival benefit compared with paclitaxel alone. Patients with advanced gastric cancer of the distal antrum or pylorus are at risk for developing gastric outlet obstruction. Traditionally, surgical gastrojejunostomy was performed for relief of symptoms and to allow continued enteral nutrition. With the advent of endoscopic stents, duodenal stenting across the obstructing tumor has emerged as a non-surgical alternative for palliation. The results of a literature review of studies evaluating gastrojejunostomy versus stenting found no differences in rate of technical success, 96% to 100%, early and late complications, and persistent symptoms. 479 Recurrent obstructive symptoms were more common with stenting. Both gastrojejunostomy and endoscopic stenting are acceptable options for the relief of malignant gastric outlet obstruction. The decision should be based on the individual clinical scenario as well as the availability of appropriate surgical or endoscopic expertise. Miscellaneous gastric tumors, gastric lymphomas, GISTs, and neuroendocrine, carcinoid, tumors are covered in chapters 32 to 34. Metastatic disease to the stomach can occur with primary tumors of the breast, melanoma, lung, ovary, liver, colon, and testicular cancers, with breast cancer being the most common. 480 other rare malignant tumors that can involve the stomach are Kaposi sarcoma, see chapter 35, myenteric schwannoma, glomus tumor, small cell carcinoma, and parietal cell carcinoma. 481-484 Miscellaneous benign tumors of the stomach include lipomas, see video 54.4, as well as pancreatic rests, see chapter 55, xanthelasma, and fundic gland cysts.